Hey everybody, welcome. I have something really exciting today to share with everybody. I don't know if you guys have seen Sherlock Rome's newest video about DNA and how it can be synthetically and artificially created. That has been true for at least 10 years that I know of. I think in the late 1970s, they started to play around with philosophy of like how that could happen. And it could have been in early 2000s where they actually were able to start doing it for sure. So this technology has been around for a while, and the concept has been around for an even longer time. So I thought I would share that today because it's an amazing topic. It's one that I've looked into, and I looked into it again briefly today, and I found this article. It's called A Beginner's Guide to Artificial DNA Synthesis. And it was written February 19th, 2025. So I found the latest and greatest in artificial DNA. And then I have some other articles that are a little bit older. One's from 2023, but still new enough to present. And I just wanted to, go over the things that they can do now in science. Like I said, science is for DNA is way beyond the 1990s education. It is so beyond that there's like ethical discussions still going on about this. However, there are those agencies that have a different set of ethical standards, and doesn't normally apply to what we understand as ethics today. But it's a fascinating topic, even just to read the discussions about it, let alone the actual science. So, all right, just give me just a second here. So this is from Azenta Life Sciences from GeneWiz. It's a beginner's guide to artificial DNA synthesis, written or published February 19th, 2025. The basics of synthetic DNA and gene synthesis. Artificial DNA synthesis, a foundational tool of synthetic biology, enables scientists to create DNA molecules of virtually any sequence without a template. Now, before in science, they needed a template to work off of. And I think that's where Sherlock Holmes left off, because he found an article where they could use anything to finish that template. Now, they can do it by manipulating molecules. They don't need those fibers anymore to make it look like a DNA strand. They can do everything independently. And so construction begins with the base-by-base synthesis of oligonucleotides, oligos, followed by assembly into double-stranded DNA, which is abbreviated dsDNA fragments. These custom DNA fragments can be used directly, cloned into vectors, or assembled into larger constructs to serve a variety of research. And it says see table below. And all the way to the left, this column says type of synthetic DNA. And then in the middle, it says structure, and at the end, it says research uses. So the oligonucleotides, they're single-stranded, and they're primers for PCR or sequencing cloned into vectors for oligos pools. PCR probes, assembly into DNA fragments. So PCR, that's something that we've heard in the Koberger case. That is how they amplify trace transfer DNA that are in minute amounts. That's polymerase cycle amplification. Oh gosh, why do I always forget what PCR is? PCR amplification. Oh my gosh. Polymerase chain reaction. Okay. PCR cycles. I'm someone who I know my acronyms, but if you ask me what everything stands for, my ADD or whatever I have just won't let me go there sometimes. So DNA fragments is the next type of synthetic DNA. That's a double-stranded helix, and it's linear. Research uses donor template for gene editing, template and in vitro transcription. Now, I do have an article called Designer Babies. This was a hot topic 10, 20 years ago when people who had the means, this is what they were doing. Plus, that was the latest and greatest in science at the time. And, you know, sometimes they can be creepy cool. I think it's totally creepy that they do that. But anyway, so they can do synthetic DNA fragments and they can assemble, there's assembly of larger DNA fragments cloned into vectors for gene expression. That's what they do for in vitro. They are manipulating gene types so that, you know, your child could have blue eyes or brown eyes or hazel eyes, in addition to different types of traits that would lean towards, you know, leadership or ambitiousness. You know, there's a whole scientific statistical standard to that. And they can also do that where they can detect certain diseases and manipulate the genes to veer away from those types of genes. So that's what they're doing with that. And the last row here for the synthetic DNA is cloned gene products. Those are double-stranded and they're circular. Recombinant protein production, transgenic gene expression, and gRNA constructs for CRISPR editing. Okay, moving down. Compared to classical molecular biology techniques like site-directed mutagenesis, gene synthesis, technology generates novel sequences with an unmatched combination of speed, precision, and flexibility. Below, we'll explore how linear and cloned DNA fragments are made and discuss what factors to consider favoring project success. DNA synthesis process. A gene synthesis process has both wet and dry lab components. Isn't that interesting? We heard, was it Dr. Gami, that was on the stand in that secret hearing, the transcript? He was talking about all of the wet and dry components when it goes into testing for DNA. And didn't he say that they did the ISP lab, was it, that did all the wet components at their lab? So a gene synthesis project has both wet and dry lab components. It can be broken down into the following steps. 1, define project goals. 2, design the frequency. 3, synthesize the oligos. Assemble oligos into linear fragments and larger constructs, if applicable. And 5, verify the sequence of the gene fragment or clone product. Okay, and then we're going to go into each one of these and what it means. So for number 1, it says define project goals. Key considerations: communications with collaborators, project timeline and planning. And you know what? these types of labs that do this, I bet you they have their own SOPs, their standard operating procedures. And anytime they get into bidding for funding or they get into other projects with other labs or wherever research wants to go, they may or may not, depending on if they're clandestine or whatever, but usually they will have that. And for whatever reason, Ortham can't give it, or the state of Idaho can't provide those. So we're still at number one, define project goals. No matter your molecular target, properly scoping your project will promote success by minimizing revisions. Be sure to consider factors like timelines for downstream array of assay work or collaborator availability, in addition to overreaching project deadlines. It may be more efficient to prioritize the gene products in a specific synthetic order or use a combination of synthetic and molecular techniques to construct them. Speed is normally critical for a gene synthesis project, but high-quality final product is just as important to reduce repeat syntheses or failed experiments. 2. Sequence design. Key considerations: literature, knowledge, and precedence. Sequence codon optimization. Once you understand the full scope of your project, it's time to map a route to synthetic success. A survey of literature will reveal known. Oh, sorry, hold on, guys. Okay, how do I... All right, that should be good. All right, where was I? Once you understand the full scope of your project, it's time to map a route to synthetic success. A survey of literature will reveal known issues in the synthesis of your target sequence and how to choose the best codons for optimized expression in your target organism. After a literature survey, further analysis, the coding DNA sequence, CDS, for critical elements such as repeats, guanine, cysteine, nucleotide content, enable unstable motifs or secondary structures, and unintended restriction enzyme sites will complicate cloning. Access to free codon optimization tool allows you to skip the literature review. It automatically improves the sequence for optimal synthesis and downstream performance in your experiments. Once optimized, the full nucleotide sequence is then deconstructed into a series of overlapping oleojo sequence. Oleojo synthesis. Key considerations: access to instrumentation, planning a synthetic route. With a route planned, it's time to roll your sleeves up and get into synthesizing. Oleojo synthesis is a highly automated process. The oleojo chain is built on solid support through cycles of chemical reactions involving precursors called phosphoramatides. With current technologies, it's not possible to achieve 100% efficiency in each cycle of reactions, leading to a subpopulation of oligos with translocations and point mutations. This issue worsens with oligos length since more cycles are needed to build the molecule. As a result, there is a trade-off between using longer oligos, which generally simplifies assembly and the sequence fidelity of the final product. So this is oligonucleotide synthesis. It looks like they have a start platform. And it goes into coupling, and there's a base. You have capping, oxidization, deblocking, And then you have your DNA sequence over here. And then over here you have repeat this many times. And you can get these DNA sequences. Wow. Okay. Let me come back here and see everybody. Need some Z's? Welcome. Jackie. Welcome, everybody. Good to see you. Oh, Need Some Z's says, I'm just joining, so I'm not sure exactly what your topic is, but I just watched a video that mentioned a study in a forensic journal from 2009 that was talking about amplifying DNA. Oh, you did? That's great that you're watching DNA videos. I do the same thing. That's awesome. Yeah, so we are talking about designer DNA. We're talking about artificial synthetic DNA, where scientists can create artificial synthetic DNA, and they don't need a template anymore. They used to need a template of some sort to get started, and then they could use whatever they wanted to manipulate the DNA to go in either direction of wherever they want it to go. Now they can do it completely independent of biological material or a template. So we're talking about artificially generating DNA. So, and we're on number 4 here. So we're talking about the steps. The DNA synthesis process. You define your project goals, design the sequence, synthesize oligos, assemble oligos into linear fragments and larger constructs, and verify the sequence of the gene fragment or cloned product. And we were down here on number 4, gene assembly. Key considerations: linear DNA fragments versus cloned DNA. And I think people are familiar with cloned. They clone a cow. I don't know how many years ago. Was that in the 90s? I can't even remember now. Well, now we're so sophisticated we can clone DNA without any template or biological material. And under my understanding, it's virtually detectable whether it's real biological material or it was synthetic and generated in a lab. So, experience with and access to plasmid vectors. Due to technical limitations, the phosphoformatide-based oligos synthesis is best to build dsDNA fragments via polymerase cycle assembly. Purified oligos are mixed and allowed to anneal to one another. Primer extension with DNA polymerase generates dsDNA, which is then amplified by PCR. And we've all heard of PCR because that's what they do to the trace transfer DNA, to amplify it, to make it bigger so we can see it. Depending on the application, the linear products can be used as is, used as in building blocks for larger fragments via methods like the Gibson assembly, or cloned into a vector. Since gene fragments are heterogeneous mixture DNA molecules, it's generally preferable to clone them prior to downstream use. Number 5, sequence verification. Key considerations: access to instrumentation, rigor of quality control based on product. Lastly, the final synthetic DNA product should be analyzed for sequence accuracy via Sanger sequencing or next-generation sequencing. We have read about next-generation sequencing. They also use that for trace transfer DNA for the minute amounts, and I do believe Ortham has a next-gen. They have this technology. They're not the only lab that has them. There are some labs out there. A lot of them have similar types, but a lot of them they're very expensive machines. They look like a copier, printer copier. They're like so big. So they have these copier machines. Well, basically, that's what they are. The PCR amplification and copier. They look like the copy machine in your office. Or, at the community office, because a lot of us work from home now. But whatever. I think you know what I mean. So they have that. We've heard that with our trace DNA. And so I do believe with the trace DNA that they find in crime scenes, they do work it up with some of this technology. And like, as we know, lately, we've had access to AI technology, which has completely changed the landscape of making YouTube videos, making songs, making graphics. There's some AI video out there that can make like a movie that is the same quality and indistinguishable from like a Hollywood movie. And that all happens in like, I think the latter part of 2024. And so just imagine how other fields and industries have rapidly increased technology-wise. And so I'm assuming, I would think that some of these techniques that they use for this could trickle down into law enforcement. Just like the AI technology has trickled over into everyday use. And I've heard that they use AI to write briefs for court. People use them to write papers or their resumes, and I get some help so that I can write some lyrics. I write lyrics for it and I prompt it, and it turns it into a song with like verses, pre-chorus, and bridge. So AI is a great tool, and so just like that type of technology that has streamed down into the public, I would think some of these behind the scenes scientific things have also trickled down into law enforcement when it comes to the scientific processes in the lab. And when you're working with transfer DNA, that's all degraded DNA. It's DNA that is not blood, sweat, semen, or saliva. So you do have to prepare some of it so that you can blow it up and look at it. And they do use software for that too. And they also do, manual comparisons, which we learned a little bit about looking at the alleles. Those peaks, those triangle, thin, tall triangle, that they do that with as well. All right, so Need Some Z's says, okay, interesting. So this sounds like a progression of amplification DNA, since that uses an initial sample that can be changed to whatever they want. Exactly. Yep, you got it. Exactly. All right, so we're going to read on here. It says sequence verification. Key considerations: access to instrumentation, rigor of quality control based product. Lastly, the final synthetic DNA product should be analyzed for sequence accuracy via Stanger sequencing or next generation sequencing. The latter can also quantitate the diversity of a library of DNA products. Restriction enzymes digests are a good complement to sequencing, especially when the vector backbone is not fully sequenced. Synthetic DNA services. The steps above can be laborious to perform in-house. Instead, to save time and focus on the downstream applications of synthetic products, consider a service provider for gene synthesis. The provider should be considered a partner in your research. As such, they should help you achieve the product of goals today and beyond. To enable your success, think about the local support, reputation for quality and reliability, expertise in synthesis and cloning, as well as ability to support long-term and multi-institution projects that have goals in preclinical and clinical trials with CLIA and GLP requirements. Key takeaways. Oh, before we get to that, so I'm wondering if Ortha has some of this technology, because they are privately owned. And, DNA cloning is kind of new, like mysterious, magical science. So I'm certain private labs like that are interested in that, especially if it's lucrative. And I would think so, because this is some heavy science here. So but I don't know for sure, but they definitely could. Key takeaways. Artificial DNA synthesis has the power to generate virtually any DNA sequence. Any. You can make it up. You can make it mimic somebody else's. Do any, virtually any. Planning and rigorous sequence verification are critical to efficient gene synthesis and project success. A service provider can dramatically reduce error, labor, and time for synthesizing genes as compared to do-it-yourself approaches. See how Cold and Optimize can improve your next synthetic DNA project by downloading our technical note on enhancing gene expression. All right, that's awesome. We just got a little lesson on the beginner's guide to artificial DNA synthesis, and this is from February 19, 2025. So now that we have that out of the way, we have a framework of what they can do now, which is virtually make any DNA sequence without any DNA templates. I have other articles here where we can get into the basics of what DNA synthesis is. Okay. Okay, so this article is also pretty new. This was written in April 29th, 2024. It's the DNA synthesis, the basics. Let me see if I can blow this up for you guys. This text is a little small. Here, okay, is it better? Yeah, it's a little better. All right, takeaway. Let me get this picture. That's a cute pic of DNA. I think my pics of DNA are way better because they're all like diamond encrusted. But this one's still cool. I like all the little, it looks like we're kind of underwater and we're glowy. I don't know. The takeaway. DNA synthesis refers to the process by which a new strand of DNA is creating using an existing DNA template. This fundamental biological process is essential for growth. Now, remember I told you anytime you're doing research, even though this article is very new at 2024, if it's this new, I wouldn't go back and try to discuss what, is newer information that has been changed, because you can use older articles. You just have to know what information changed from there and know which information stayed the same. So in this article, I wouldn't go back and try to see if there are newer articles that would make this information change a little bit. But even this article says using existent DNA template. But as we just learned, we don't need that anymore. They can do it without that. This fundamental biological process is essential for growth, development, and maintenance of living organisms. DNA synthesis occurs during various biological events, such as cell division, DNA repair, and the replication of genetic material. Now, our bodies automatically do DNA repair and they automatically do cell division. There's different processes that are already built in. They are trying to mimic those processes in the lab. And by studying how our body operates, that is what they were able to incorporate into the processes and what they needed to recreate to be able to do this. So synthesis occurs during various biological events, such as cell division, DNA repair, and the replication of genetic material. What is DNA synthesis? During cell division, a cell must duplicate its DNA so that each cell receives a complete set of genetic instructions. DNA replication involves the synthesis of a contemporary DNA strand for each of the original DNA strands. The process follows a semi-conservative model where each new DNA molecule contains one strand from the original molecule and one newly synthesized strand. DNA is double-stranded and includes sense strands, code in the 5 to 3 direction, and antisense strands, the non-coding DNA strand of a gene. And I think side note, we recently read an article where this non-coded DNA or the cell-free DNA, it's meant, I think, to detect cancer and other diseases when the cell doesn't have a nucleus, or this non-coded one. And police are using that because it's easier to identify someone that way. And I'm not sure if there's a connection there with this, where you can manipulate that cell-free, that nucleus-free non-coding DNA a little bit more. You can manipulate it to match your suspect's DNA. And that brings me to another side note. So typically we've always been taught that like they find the source DNA at the scene, and then they have like a list of people that they compare it to that. Nowadays, I think it's reversed. They have a DNA profile from the scene, and then they pick a suspect they like, and then they do these little things to build it up and make it look the most it can to whoever their suspect is. I just think there's different techniques that they use now. That one doesn't seem quite right, but I do think we're seeing that more and more now. Anyway, so back to the article. The key steps in DNA synthesis include initiation. The process begins at specific sites on the DNA molecule called origins of replication. Enzymes called helicases unwind and separate the DNA strands, creating a replication fork. Primer synthesis. Primers, short RNA sequences, are synthesized by an enzyme called primase. Primers provide a starting point for DNA synthesis. DNA synthesis. DNA polymerase enzymes catalyze the addition of nucleotides to the growing DNA strand. Nucleotides are the building blocks of DNA, and they pair with complementary bases on the template strand, adrenaline with thymine, guanine, and cysteine. DNA polymerase adds new nucleotides to the 3 end of the growing strand. Now, I don't know if the strand is actually growing, or if they just mean because you're working with it, it's getting, bigger and longer. I was briefly reading this before, and I can't verify it, but someone on Reddit was saying it doesn't actually grow, but I don't know about that because, I would have to dig in a little bit further, especially with these scholarly journals or something a little bit more reputable than Reddit. Reddit is okay to read as long as people cite their work, or you can take their general idea and plug it in and see if there's any scholarly journals that actually explain a little bit more what they're talking about. So you just have to be careful with Reddit, do a little bit more work when you look at those types of things. But somebody on Reddit said it doesn't actually grow. I don't know how true that is. I would have to read more about it. They are using the word growing here. Maybe they'll tell us a little bit further in the article whether it actually does grow. So, proofreading DNA. Oh, wait, proofreading. That is so funny. You had to proofread DNA. Proofreading. DNA polymerase also has proofreading capabilities to ensure accuracy in base pairing. Incorrectly paired nucleotides are removed and replaced with the corrected pair. Ligation. After the synthesis of new DNA strand, the fragments, I'm assuming this is a person's name, and maybe they developed a technique. Okazaki fragments and the lagging strand are joined together by enzymes called ligases. DNA synthesis is a highly regulated and intricate process involving various enzymes and accessory proteins. It plays a crucial role in maintaining the integrity and stability of genetic information in living organisms. All right, let me come back and see what you guys are saying here. Oh, we have a Heather Fitzgerald in the house. Hello, Heather. Welcome. And hello, Pip Pot. He's so cute. Okay. Heather Fitzgerald says, wouldn't it be crazy to find out Brian donated bone marrow? That would be so crazy to find out. That would answer some questions as well. Heather Fitzgerald says, yeah, like the lab lady in Colorado. Right? What was her name? Melissa or Missy Beavers or something? Yeah. Yeah, I don't know. What's that experiment? I forgot. I should know this off the top of my head. Where they had prisoners and inmates, and like by the end of the study, psychologically, they kind of these roles imprinted on these people, and so it really did turn in to the prison guards. They really psychologically took their role as having power over these inmates, and these inmates, it was psychologically imprinted on them that they were inmates. And I think, did they start fighting or something? I think it's that same process where now they're taking, the law enforcement is taking the source DNA from the scene, and they have it, they build it up a little bit, they have their list of suspects, and they build up the source material to match one of their suspects, instead of the other way around where it's supposed to be you honestly and ethically and according to procedure source the DNA from the crime scene and then see if it matches anyone else. Do you know there's a little bit of a distinction there? Aww, Heather, Pip-Pot. He sat up when I said hello. Oh, he's so cute. Okay. Heather Fitzgerald says, yes, it has imprints on LE. Yeah. All right. So then reading on. Hello and welcome, everybody. What is DNA synthesis used for? DNA synthesis performed outside of a cell has various important applications across different fields of science and technology. There are two types of synthesis, natural and synthetic. Synthetic DNA synthesis is a technique that allows scientists to create DNA molecules without a template and in almost any sequence. That's what we read in the article earlier. But now they can create DNA molecules in any type of DNA strand without a template. Some of the key uses of DNA synthesis include research and biotechnology, gene cloning. DNA synthesis is used to create copies of specific DNA fragments or genes to further study. And you know what? I think trace transfer DNA are DNA fragments because it's damaged. It's not a full profile. So they have little bits of it, and they might have some missing pieces here and there, or they might just have a handful of fragments that aren't in order. So there is a whole research industry out there to clone those fragments. And I guess is that essentially what you're doing when you do the PCR, the polymerase chain reaction amplification process? All right. Moving on. Site-directed mutagenesis. Researchers can introduce specific mutations into a DNA sequence for further studies. First thing that comes to mind, side note, when I read that is they might be able to introduce maybe different pharmaceuticals or different scenarios or different processes that the body already does into cancerous cells to see how it reacts. And I was reading months and months ago that they were trying to figure out how the osmosis, the autosis, the cell death that cancerous cells go through, And so they were trying to find some sort of pharmaceutical that would reverse that process. But in order to do that, they needed to be able to reproduce the conditions and what causes that and what chemical reactions are there. All right, back to the reading out. Constructing plasmids and vectors. DNA synthesis is crucial for creating customized plasmids and vectors used in genetic engineering and molecular biology experiments. Genome editing, CRISPR, Cas9 technology. Custom DNA synthesis is integral to the design and synthesis of guide RNAs and DNA repair templates for genome editing applications using CRISPR-Cas9 and other genome editing techniques. DNA sequencing. Sequence controls. Synthetic DNA is used as controls and standards in DNA sequencing experiments to validate the accuracy and the sequencing process. Artificial DNA synthesis permits the identification of novel biological systems. Sequencing is used for confirmation activities. Diagnostic applications, probe synthesis. DNA probes used in various diagnostic techniques like fluorescein, in situ hybridization, FISH, or polymerase chain reaction, PCR, are often synthesized to specifically target and detect particular DNA sequences. They do that. These two techniques they use in criminal identification. Sometimes they do inject the trace transfer DNA with like a fluorescent liquid. Kind of like how you have to drink that like toxic yellow liquid before you get like an MRI or whatever, so they can look at your insides. I think they do the same thing to these trace amounts of DNA to make them more visible. So for criminal identification, they are doing both of these, which is the fluorescent in situ hybridization FISH, or something related to the fluorescent use of injecting the genetic material. And I think we've all heard the PCR process. All right, and also the next one is vaccine development research. Antigen synthesis. DNA synthesis is used to create synthetic genes that encode antigens for the development of vaccines, which I think that's what they were doing in that cancer research project, to understand more about the different functions that the cancer cell goes through, and even before it gets to that point where it goes through the death cycle, to understand different parts of the way before it gets there as well. So DNA fragments and genes can be used in vitro transcription templates for mRNA production. We will also do a live on RNA. There has been a lot of research considering that lately. There's a lot of it. So we'll have to get into that too. And that is, it's rivaling DNA because there's so much information out there. Before, there wasn't really any information on RNA at all. All right, so therapeutic proteins, recombinant DNA technology. This involves inserting DNA that encode the protein into bacterial or mammalian cells, expressing the protein in those cells and then purifying it from them. Antibodies, detection. Antibodies can be used to detect newly synthesized DNA. That is awesome. They have antibodies that can go in there, do their thing, and they can figure out, is it synthesized or what? That is awesome. That's so cool. I would like to see that. They're like little worker bees and they have a job to do. All right. Reading on. Drug discovery research. Library synthesis. DNA synthesis is employed to generate libraries of DNA sequences, which can be screened for potential drug candidates or used in high throughput screening arrays. So what that says to me is when they're doing this drug discovery research, they must have a DNA database that they use to see which one of our DNA would be good or is the target type of DNA for them to do their drug trials. That's just what immediately comes to my head. I don't know if that's true though. Or used in high throughput arrays. This is just one example, but there are many more, including protein engineering and CRISPR library screening. Synthetic biology, designing synthetic genomes. DNA synthesis plays a crucial role in the field of synthetic biology, where researchers design and engineer novel organisms with specific functions or traits. And there is also new technology in pharmaceuticals where they are starting to do that, where they're starting to make pharmaceuticals that mimic specific functions or traits to help with different diseases like diabetes and cancer. Hold on a second. Okay. Next one, personalized medicine, custom DNA constructs. In personalized medicine, this is what it's called. This is what I was reading about, personalized medicine. In personalized medicine, synthetic DNA can be used to create custom therapeutic constructs, such as personalized gene therapies or gene editing tools tailored to individuals. That was what it was called. I didn't remember it until now. Yeah, so reading on here, it says functional genomics, synthetic genes for functional studies. DNA synthesis facilitates the creation of synthetic genes for studying their function or regulatory environments and cellular processes. The last one in this list is academic and educational purposes. Teaching and training. Synthetic DNA is often used in educational settings to teach molecular biology concepts and techniques. So that list that we just read here, I'm going to go back and scroll up, is called What is DNA Synthesis Used For? And so we started out with gene cloning under research and biotechnology, genome editing with CRISPR, DNA sequencing, sequencing is used to confirm activities and for the accuracy for the vaccine development research, therapeutic proteins, antibodies, drug discovery research, designing synthetic genomes, personalized medicine, which I was particularly interested in. They have functional genomics, and the study of the function of regulatory elements of cellular processes. That also goes into personalized medicine. And of course, always academic and educational purposes. So that was just a brief overview of what we just read there. And let me come back and see what you guys are doing in the chat. Hello, Truth and Justice, True Crime with Anna D. Welcome. Heather Fitzgerald says, really makes me wonder all those times that Murray Povich said you're the father. How many of those are accurate? Hey, if their ratings were down, I mean, who would ever question it, right? It's kind of like how it's like, who would ever question law enforcement? Is Miss Potts in here too? Did I miss it? I saw your comment on Miss Potts' post. Oh, timing is everything. Yeah, I would agree. You're allergic to that dye that they inject you with? It looks like toxic, like, waste from, like, you know, a nuclear power plant dye that they inject into you when you do MRIs. I can't believe you're allergic. Have you ever had to have an MRI done, Heather? Do they not use that highlighter dye? I do so good. It's so good to see you, Anna. Heather Fitzgerald says, literally after over 10 years of having MRIs, all of a sudden one day I was allergic. It was crazy. I'd say it was absolute close to death I've ever been. Oh my gosh. I can't even imagine how that was like. Were you in a hospital setting when that happened? Or like just a clinic? That's so scary, Heather. I'm so sorry about that. Well, at least you know now. Heather says it was crazy. They could see my throat was closing in the MRI machine. Oh my God. I can't even imagine. You poor thing. Well, they're going to have to find something else for you. Heather Fitzgerald says they had to stop the MRI and rush me to the other side of the hospital. Oh, good. At least you were in a hospital setting. That's great. Good. Oh, gosh. Okay. Let's get back to the article here. It says, how does IDT, which this is the organization, we're reading their article. So they're called Integrated DNA Technologies. So how does Integrated DNA Technologies support DNA synthesis? Integrated DNA Technologies, IDT, is a leading provider of products and services related to DNA, including custom DNA synthesis. The general methodology for DNA synthesis involves chemical synthesis on solid supports. Gene synthesis from IDT is an easy solution for researchers who want to bypass in-house cloning and instead move right into functional studies using 100% sequence verified clonal DNA. Bypass in-house cloning. Why? Are there like standard procedures where it's like we're not gonna, engage in cloning, but then we're like, hey, just, contract out those services to another lab that'll do it for you. And they already have 100% sequence verified clonal DNA. They already have it for you. They already have the DNA that's artificially made. Okay. Ordering. Oh, you can order it. It's on demand, guys. Do you guys need to order some synthetic artificial DNA? You can. Yeah. So one of the reasons why I wanted to get on here is because somebody try to I guess debunk one of Sherlock Rose's videos, but Sherlock Holmes is 100% correct. So I don't know who that guy is that was like, oh, I looked at Snopes and New York Times, and they say that, creating artificial DNA is not possible. Oh, did I have a lot to say about that? So that's kind of why I'm here today. And I know that particularly I was interested in the type of synthetic DNA that they're using for the personalized methods for pharmaceuticals. And so now we're at the point where you could order your own cloned or synthetic DNA and deliver it right to your doorstep. It says ordering. There's a link. Can I put some in a cart and order it like Amazon? This says ordering from IDT is easy and includes guaranteed delivery dates. There's no minimum order requirement or additional charges for custom vectors. All right, so I guess you could order like pre-made, because I was thinking about getting a microscope and ordering pre-made slides so that we could all look under the microscope. So I guess they have pre-made for these cloned DNAs for you to do whatever research you're doing and whyever you're ordering this synthetic artificial DNA. But you can also get it to them to create whatever you want instead of those pre-made stuff. Oh wait, did I click? I meant to click back. All right. Yeah, just like DoorDash. You want to order some synthetic DNA from DoorDash? Why not? DoorDash delivers more than just food, right? Don't they get a Home Depot now too? Why not just put synthetic artificial cloning on there too? It's like I'm conducting experiments on DNA. Leave me alone. Heather Fitzgerald says, then I had a horrible shingles outbreak days later. I guess it was due to the stress on my system. Yes, I had the chickenpox as a child. Wow. Yeah, I'm sure that was a whole shock to your system. David Bird, welcome. David Bird says, special rates for 3-letter agencies. Right? Right, they do. They probably get a discount. Look, I accidentally clicked on the ordering page. Let me see if I can zoom in. Let's see what we could order. You know how like on the DoorDash, you can like hit like request for this restaurant, you know, to be on DoorDash. Can we hit request for these guys? Introducing our newly enhanced, I'll go back to the article, but we're taking a detour. Oh, look, this 40% off. Hold on, guys. We're taking a detour. Introducing our newly enhanced gene synthesis. New. This easy-to-use solution is for researchers who want to skip the in-house cloning steps and move quickly into functional studies with 100% sequence-verified clonal DNA. All right. So it says 40% off additional options for genes. What? Gene add-ons? What? What are we doing? What are they doing? Take 40% off new gene synthesis enhancement features, including glycerol stocks, transfected grade preps, and endotoxin-free preps. It expires March 31st. There's like a week. And you can also get $500 off when you onboard your own vector. All right. And that applies to first gene synthesis order. After onboarding, promo credit will be issued upon completion of the IDT vector onboarding. All right. So, under ordering, the first thing it says is complete synthetic genes with 100% sequence verification are delivered in a cloning or expression vector and are ready to use for a variety of applications. Never any reoccurring or hidden fees for custom vectors. Transfected grade endotoxin-free options available, but those are add-ons, right? You got to pay extra for those. Oh, no, but it says, no, I think you have to pay extra. Guaranteed delivery dates. Oh, okay, genes in tubes. We got biological material in the tubes, guys. You can order those. No order minimum for tubes. Delivered dry. Normalized to 4 UG with scale-up options available. Glycerol stocks available. You can do a mini gene. I guess this is quantity. Oh, well, if you get the mini gene, it only takes 8 business days, and then it gets longer from there. Gene in plates, available in PCR and ectoplates. It's 3 UG, just delivered dry or wet, normalized to 2G. So I got a question for you guys. If this, hi Preston B, welcome. Yes, let's DoorDash some DNA. I got a question for you. So it looks like this company will DoorDash you some DNA. They must have some ideal environments to be able to transport this biological material. I mean, they're selling test tubes. Why couldn't MPD do that? They had an officer who is not trained in preserving the environments needed for biological samples to travel. So that DNA sample is most likely compromised. You know, it's kind of like when you rent a storage unit. For some things that you're storing, you need that climate control. You know. So MPD was it was ISP, I'm sorry, it was ISP. They're completely unqualified to travel with DNA. So that DNA was compromised even before it got on a plane. David Bird says, NB partial samples will double in size. NB, what's that? David Bird, can you please give us a little bit more detail on your comment? All right, so guys, it's 40% off right now, but we only have until July 31st. Daddy Kane, welcome. We're talking about artificially creating DNA. Note, partial samples will double in size. Well, yeah, because partial samples will have to double in size, right, for them to complete it? Okay, so David Bird says, note, partial samples will double in size. I think that makes sense because they have to like blow it up and change it and try to make it full. So this is what I was talking about earlier. So they have apparently, maybe, I don't know if we're even sure anymore, that they had the trace transfer from the inside of the snap, maybe they cut the female part of it. Partial and ambiguous, right. And so this is what I'm talking about. They will build it up, they'll build the source DNA up, but in that process, they'll try to make it look like whoever their suspect is. It's like backwards. Like you would think they would honestly and ethically, source the DNA from the crime scene and then use that to see who it matches. But no, I think nowadays they're building up that sample and then they're matching it as close as they can to whoever their suspect is. And yeah, partial ambiguous. Yeah, Becca Barlow would not have used those words if they weren't true. She's not going to put in there thing that would like hurt her career or just flat out not true. Those things are very true. It was partial and ambiguous, which makes sense. Danny Kane says, do you think they built it out of using his father's DNA? Anything is possible, Danny Kane. I think they did something weird to the DNA. I mean, because everything else, okay, this is the thing with trace transfer cases. The trace transfer cannot be your only piece of evidence because that usually would indicate this person is innocent if there's no other connection. The way that trace transfer may have a connection to a case is if there's other factors that connect that suspect to that crime, but we don't have that here. So it is quite possible something happened to the DNA. I don't know what, things may not have happened to the DNA. I just have a feeling that's the practice now, like across the board, not just specifically this case though. David Bird says his father's or even his, both. And I think it falls into the category of confirmation or the bias. Anytime you read discussions about DNA, especially transfer DNA, and like standards and like how it should be read and, what are the best practices. They write best practices, but even on that level, work culture can change that depending on what the personality of the environment you're in. That can make things, sociably okay, where it goes against whatever is ethically or in the standards, just because that naturally happens over time. Like I was saying, that experiment with the prisoners and the prison guards, where something psychological happens when you're in a vacuum of an environment, like a work environment. All right, back to ordering some DNA. So you can order genes and plates. The mini genes only take about 8 days. Well, let's read these like disclaimers here. Sometimes they can be interesting. So star. Gene sequences with added complexity can interfere with assembly and or sequencing performance. Such sequences may result in less synthesis, yield, or additional service charges. The time required to manufacture a gene is dependent on length, complexity, and vector choice. 85% of orders are ready to ship within a number of business days as indicated. Sequences that are unstable or toxic to E. coli can substantially affect delivery times. Echo is trademark of the LabSite Inc., a Beckman Coulter company. Plates with less than 24 fragments will incur an additional fee applied at checkout. Need your jeans faster? Click here for our new rapid jeans. Oh, we're going to click on that. Because look, I mean, if they are going to make this synthetic DNA and it takes about 8 days to ship, I don't know. That's kind of fast. They have even faster ones. All right. Additional options for jeans. Jean glycerol stock. That's $25 a jean. Okay, it doesn't have additional tat or yield. You have transfection jean or transfection grade mid-scale prep. That's $120 a jean. Additional time is 3 business days, and the yield is 100 UG. Then you have the exotoxin-free mid-scale prep. That's $160 per jean, and that'll take an additional 4 days, and the yield is also 100 UG. Oh, they have one of these fancy disclaimers. It says vectors identified as low copy vectors will have a reduced yield of 50 UG. This will be determined during the custom onboarding process. Now, I'm not saying necessarily that the Idaho State Police or Moscow Police did this to the DNA. I'm just simply responding to Sherlock Holmes or Sherlock Holmes and saying he was 100% correct when they can artificially make DNA. And not only that, they'll ship it in about 8 business days. So, and then I'm just commenting on the case as we're going through here to kind of relate it to what we've all been talking about. But I am not by any means saying that they did this to break Koberger's DNA. What I am saying is there could be a work environment, where bias is that could contribute to not so great standard practices or operating procedures. Now, would that affect the biological samples that they have? I don't know. That's all I'm really saying there. And if the DNA was actually manipulated, I have no idea. But I just think this is fun. Okay, just so that we have that disclaimer out there. All right, so this is IDT vectors. IDT has several catalog vectors available for use in your experiments, but if you don't see one here that fits your needs, see custom vector onboarding below. We have several options for cloning vectors and vectors ready for protein expression experiments in both bacterial and mammalian cells. When selecting our standard cloning vector option, if your insert is less than 250 bases, we will use the PIDTS Smart Vector. All other links will be cloned into our PUCIDT gate with antibiotic selection marker of your choice. We also offer vectors that have been optimized for use with Golden Gate assembly. They will not have the most frequently used type ISS restriction sites. Upon receiving your gene, you can then subclone into the vector of your choice using a variety of methods. The identity of the cloning vector used is sequenced and inserted. An insertion site will be confirmed in the documentation that accompanies your product. And in this table, it says best fit vectors for genes. Okay, so look, certain vectors, their application, half of them look like they're for cloning. One of them looks like mammalian expression, and the other ones look like bacterial expression. So they, depending on what kind of project you have, that'll determine what types of genes or DNA you need to experiment on your project. Now, what do they mean by custom vector onboarding? If your experiment requires a vector that isn't in our standard genes tool, no problem. We can easily onboard your own custom vector through our website without requesting a quote. Advantaging to onboarding your vector with IDT saves you time, and we do the cloning for you. Once onboarded, you can order cloned genes in your vector anytime. Oh my gosh, like once Sherlock Holmes hears this, you can go back to that guy and be like, you can have DNA shipped to your doorstep. This is readily available. They have an eight-day shipping. Wow, it could be 12 or more depending on how custom your vector is. Okay, so advantages of onboarding. Saves you time, and we will do the cloning for you. Once onboarded, you can order cloned genes in your vector anytime. Onboarding has a one-time cost of $500. That's a deal. My God, that's so low. I thought that would be way higher. No reoccurring fees for additional inserts. You know, there's no monthly subscription fees. It's just one time, $500. And then your work is on there and you can order it anytime. Fast turnaround times. Confidentiality is a high priority to ensure your IP is protected. What is that? Intellectual property? What is IP? Oh God, look, they have little icons on the whole process. Oh, Tanel Ray, welcome. Oh, Tanel Ray says misusing the PCR process. That is a big one. That is a big step that is manipulatable. Yes, good call. Let me see if I can make this chart bigger. All right, everybody take a screenshot. This is how you upload your own vector, which is your own DNA, your sequence. Look, number 1 on the computer. Submit your vector sequence in our onboarding tool. Then get your credit card out. Purchase confirmation and vector shipping instructions will be sent. They will clone whatever you put up, in the onboarding tool. And look, they have like this test tube. They'll clone DNA in the test tube and they'll ship it back to you. And then quality control, a manufacturability test. Okay, so they have very high standards. They have that. They're gonna do that after they pack and ship. Yeah, well, they're gonna make sure before and after they have high quality control. Number 5, they'll mail you or email you notification of the onboarding results. Awesome. Everybody get a screenshot of this, of how you can order your own custom artificially cloned synthetic DNA. All right. Okay. And then down here it says request a consultation. Have questions for our experts? Want to onboard your own vector? Your time is valuable. I will prioritize your inquiry. Click on Request a consultation to provide brief information about your project. I will be in touch to discuss it ASAP. I am joking. But they got Brian Koberger's DNA from his car handle or whatever. They sent it to a place like this so they could make the artificial DNA. They're like, yeah, make this specimen to be this. I'm totally joking when I say that. I can't stop but laugh halfway through. But wouldn't that be something if they ordered it through DoorDash for Brian Koberger? Product details. Genes and mini genes are next generation certified. I mean, wow, they're like accredited stuff. This is, I don't know, we found top of the line synthetic DNA. Like this looks like a really legit company. Circular double-stranded DNA in a plasmid DNA sequences, 25 BP to 5 KB, are provided with IDT's in-house vectors, expression vectors, or a custom vector of your choice without additional coding fees. Sequences greater than 5 KB can be reviewed for acceptance as a custom project. Plasmids are delivered ready to use in your preferred configuration with accompanying quality control report. Storage conditions. You can do it short term at less than 2 weeks at 4 degrees Celsius, and long term up to 2 years, you want to do it negative degrees Celsius. So the ISP guy who flew with the DNA, was it from the trash in Pennsylvania? Or any of them? Was it climate controlled when you moved it? Product data. Sequence verification. All inserts for genes and mini-gene products are sequence verified by next-generation sequencing. Upon delivery of the plasmids, a next-generation sequencing report, plasmid map, and FASTA file are all available to you in your online IDT account. You know how like DoorDash is like, you can just reorder like past orders. You have an online account with them, and you can just keep hitting reorder. They save your order for you. All right, resources. They have a DNA cloning guide. There's a lot on it. I don't know. I guess you have to have an account with them. And then they have antibody discovery ebook, driving antibody discovery with synthetic biology, protein engineering booklet, advanced possibilities of DNA engineering, the mutagenesis handbook, web tools, gene and gene fragments order status, codon optimization tool. They have flyers, antibody discovery infographic, antibody discovery product flyer, genes and gene fragments flyer. They have videos. We can't click on the DNA cloning guide. I clicked on the DNA cloning guide. It's locked though. Oh no! Oh my god! Oh look! There's a DNA cloning guide you can download for if you need to order it. They'll help you through it. What do you guys think of that? So it did let me click on it. Okay, what's inside the DNA cloning guide? Learn about common DNA cloning techniques. Understand important considerations for DNA sequence design. Troubleshoot issues that may arise when conducting your cloning experiment. Fill out the form to download your own copy. Wow. That's pretty awesome. Well, download a copy and send it to that guy who's severely misinformed. All right. I want to get back to this is it. All right. We took a detour about ordering your own DNA. That was very beneficial to learn about that. And then right after this live, Heather Fitzgerald, she is going to go on DoorDash. And you know how you can request restaurants to be on DoorDash? She's going to go request this company. I'm just joking. She's not going to do it. All right. I accidentally clicked on ordering, so this is where we left off. Gene synthesis. We're going to read a little bit about how IDD support DNA synthesis. Gene synthesis from IDT is an easy solution for researchers who want to bypass in-house cloning, instead move right into the functional studies using 100% sequence verified clonal DNA. Ordering from IDT is easy and includes guaranteed delivery dates. There is no minimum required or additional charges for custom vectors. IDT leverages automated synthesizers and advanced technologies to carry out these steps efficiently, sustainably, and with high precision. The ability to synthesize custom DNA sequences is crucial for various applications, including research, diagnostics, and biotechnology. Yeah, if you subscribe, their blog is delivered straight to you. Oh, that was it? Okay, well, what happens? Oh. Let me come back and see what you guys are saying. Okay. Well, that was awesome. Okay, so I do have another article here from Harvard. This one I do believe is a little older. This is from the Petrie Flom Center at the Health Law, Policy, and Biotechnology and Bioethics at Harvard Law School. This one is under their assisted reproduction technologies section, and it's called Designer Babies, the Ethical and Regulatory Implications of Polygenic Embryo Screening. So you can create DNA using, manipulating molecules and cells outside the cell. And as we know, if you have enough cells and enough DNA sequencing, you can make a baby. And let's read more about that. Look at this. You guys see this artwork? I'm also really interested in looking at DNA artwork. Look, they're splicing in a DNA sequence right here. That's funny. Okay. All right. Well, this isn't that old because it says in here December 2023. I don't see a date on here. Let me just go to the bottom. Well, it's copyrighted 2025. So this is a newer article. This article is Designer Babies, the Ethical and Regulatory Implications of Polygenic Embryo Screening. Okay, written by Hannah Raham. New technologies are increasing the accessibility. Let me see if I can make this a little bigger for you guys. All right, is that big enough? Yeah. All right. New technologies are increasing the accessibility of polygenic embryos screening, which can assess the likelihood of an embryo developing polygenic diseases, for example, diabetes, schizophrenia, or provide insight into certain polygenic traits, height, intelligence. This is what I was telling you. When you have a designer baby, you can influence the genes to stray away from diabetes and schizophrenia, and they can provide insight for you for height and intelligence and all the good things that they want, ambition, all of these good qualities that, that could be linked to genes depending on people's personalities. Somebody who's a leader, somebody who will be successful, somebody who has drive, passion. To design your child, you can also just design their genes and, what kind of intellectual abilities they could possibly have. This procedure has many complex clinical, social, and ethical implications, but is currently unregulated in the US. Oh boy. All right. ORCID, I love that name. ORCID Biosciences and Polygenic Embryo Screening. In December 2023, the startup company ORCID Biosciences launched a genetic testing service that enables prospective parents undergoing in vitro fertilization to screen embryos and identify genetic predispositions to common diseases. Prospective parents can then select the most desirable embryo to be used for pregnancy. And I'm wondering if these embryos that they are selecting, or this company has ever outsourced to a company like IDT that we were just reading about, where they can genetically, synthetically create their own DNA. There's and especially now that DNA science has gotten to the point where they can reproduce functions of the body for personalized medicine, so that, like, according to your individual needs and your individual DNA, they can make a medicine that help you fight the cancer or anything like that. So they are also able to do that, so that parents can go in and pick their embryos with guided with guided help from these different companies that put their standard operating procedures on how they actually do that. Okay, so to screen embryos and identify genetic dispositions to common diseases, prospective parents can then select the most desirable embryo to be used for pregnancy. ORCID claims that it can sequence more than 99 of the embryo's genome to test for both monogenic diseases caused by mutation of a single gene and polygenic caused by a combination or caused by the combined action of multiple genes. So it looks like ORCID can do the synthetic DNA in-house, is what it sounds like here. Because ORCID claims it can sequence more than 99% of the embryo's genome. So yeah, they're making a designer baby. They are manipulating the DNA. So to say that that artificial DNA is not here, no, it's here and it's been here a while. Where they are now with it. And I'm sure that there's scholarly studies and journals that are, working and studying on things that are even more technically advanced than this as we speak. They're probably in trials now to see what works and what doesn't for, whatever is going to come down the pike in the future. So ORCID does claim that they can sequence more than 99% of an embryo's genome to test for both monogenic diseases caused by a mutation of a single gene and polygenic diseases caused by a combination or a combined action of multiple genes. ORCID's testing service has sparked debate about the utility and consequences of polygenic embryo screening. Yeah, so not only do they have these types of studies, these types of companies, these types of entities that are doing all this, even just reading about the discussions back and forth about the ethics of this is very interesting too. There's some points that companies or people have that I never would have thought. And there's some I agree with, I don't agree with, but I find it all fascinating. I just absorb all the information. All right, moving on. Until recently, implantation genetic testing of embryos was limited to severe life-limiting monogenic diseases, such as cystic fibrosis or muscular dystrophy. ORCID's testing for common polygenic diseases, such as coronary artery disease, diabetes, and bipolar disorder, is more controversial, in part because of the technology or the technological complexity of predicting a condition arising from multiple genes. So it sounds like this technology isn't perfect. They might actually have AI that can run these complex algorithms quickly to see how close they can get to predicting and sorting out the negative aspects of what they don't want in their synthetic artificially generated genome. So that sounds like to me. In part because the technological complexity of predicting a condition arising from multiple genes, which results in a low predictive power of polygenic scores. Researchers and professional biomedical organizations have expressed concerns about polygenic risk scores, such as the fact that they do not determine whether someone will develop a condition and only measurable possible risks. Also, polygenic risk data are not specific to one condition. Action for one condition can affect other genetic traits. Despite these concerns, recent research found that 72% of the American public approve of polygenic embryo screening, and 82% would be interested in using polygenic embryo screening if already undergoing IVF. See, isn't that interesting? And this article is good in that way, where it's talking about the science, it's talking about the startup company, it's talking about the options, but it's also bringing in the discussion of like how people feel about it. What are the ethics behind this? Is this okay? So they are weaving that into this argument because it's always a part of the conversation, especially when you get to controversial science like this. So reading on. Currently, companies like Orchid only offer polygenic embryo screening for disease conditions, but polygenic testing methodologies can be used for non-disease traits. Would that be like eye color, hair color for non-disease? I don't know. I'm sure those are easy to do. Public approval for polygenic embryo screening is much higher for physical health conditions. For example, prostate cancer. 77% of the psychiatric health conditions, for example, Alzheimer's, 72%. Then for the behavioral traits, for example, intelligence, 36%, and physical traits like height, 30%. All right, so okay, so this is the public approval. So they approve of health conditions, that's 77%. And for psychiatric health conditions, what is that? The public approves it at 72%. And for behavioral, like intelligence, so you're a leader, you're powerful, you have all those things to be successful, they agree, the public agrees for about 30%, 36% think it's okay. And then when you're doing it for physical traits, the public says 30% is okay. So am I reading that right? Public approval for screening is much higher. Okay, yeah. So they did put that in there. All right. We're reading on to ethical implications of the polygenic embryo screening. Polygenic embryo screening engenders complicated and challenging ethical considerations. The procedure is expensive and not covered by insurance. Ooh, I wouldn't think it would be. I've never seen an insurance claim for that. Orchid's genetic test will cost consumers $2,500 per embryo. I feel like the cost of that has come down a lot because this technology has been around for a very long time, but it was very exclusive, 10, 20 years ago. Not including the costs of IVF. Oh, it's an addition. Okay, well then, yeah, the costs are still high. Which has led to concern about perpetuating wealth inequalities. Exactly. The wealth inequalities here. And that's probably why it's not mainstream. It's not public. The public doesn't really know about it. This type of information is not readily available unless you look into it like I do. I read all this stuff. So, yeah. So for that guy to try to debunk Sherlock Holmes, it's like, this just isn't your world. So you don't know that it does exist. I don't think he's ever going to get into this. But this has been around for a long time. And the ethical implications, they're lessening. But can you imagine 10 or 20 years ago, this would have been like a shock to people. So this wouldn't be public knowledge. Or if it was very hush-hush. So, reading on. In the words of the philosopher Peter Singer, The present generation of wealthy people will have the opportunity to embed their advantages in the genes of their offspring. See, this has been something that the wealthy has wanted to do for a long time. It's been around. Let me read that quote again. It kind of sent chills down my spine here. The present generation of wealthy people will have the opportunity to embed their advantages in the genes of their offspring. That is genetic cloning, genetic synthetic DNA. Although it doesn't explicitly say it, that's exactly what that means. Relatedly, the option to create a desired baby by selecting an embryo based on a variety of screened characteristics can lead to a consumerist approach to reproduction, which can have consequences for the parent-child relationship. For example, people have expressed concerns about hyperparenting in the selection of embryos based on certain traits. If polygenic embryo screening is offered for behavioral traits, some parents may select an embryo based on traits that value, like athleticism or academic achievement, and impose pressure on their children to pursue activities in line with these traits. Whoa. Wow. See, and this has been around so long that they have studies, which, I think you can infer that would happen naturally, that if, you bought your desired baby, that you would want to, guide that baby on like how you picked it out. So I think, in psychology, that wouldn't be a stretch to think, but they probably have lots of psychology behind these. Because, sometimes when you sign up for something or like a new medication, they want to keep track of you because you're in a study. That maybe the first couple of embryos that were created, they were monitored. I mean, this is like straight from a sci-fi TV show, but this is all real. Polygenic embryo screening is also susceptible to an expressivist critique that selecting against certain traits may imply a lower worth of individuals who possess selected against traits and can increase stigma surrounding those traits. This critique can be rebutted by arguing that screening is a preventative health measure and that preventing the incidence of a condition does not imply a negative view towards individuals with that condition. For example, vaccinating against a disease does not make a negative judgment about individuals with the disease. The expressivist critique is related to concerns of disability rights advocates that screening embryos for disabilities results in a harmful view of disabilities. Their concerns span beyond attitudes towards individuals living with disabilities and instead focus on the argument that screening reinforces the mistaken belief that disability itself is a problem to be solved. Many disability rights advocates argue that the negative aspect of life with a disability are attributable to societal discrimination rather than the medical condition itself. They therefore see disability as just another form of human variation, like sex or race, and not something to be prevented. Additionally, while embryo screening may determine the risk of a child experiencing particular disability, it cannot predict a child's quality of life with that disability. People without disabilities often perceive the quality of life of those with disabilities as much lower than it actually is. Many people with disabilities report a similar quality of life to people without disabilities. Embryo screening for disabilities thus often does not reflect the views of individuals living with those disabilities. regulating the polygenic embryo screening. All right, before I get into that, let me come back and say hi to everybody. Heather Fitzgerald says, are they trying to make test tube babies? That's exactly what it is. They've been doing that for a very long time. Yes. And, you know, it goes right in line with this designer synthetic DNA. That's the next evolution of that. That's where that can lead. If it doesn't, a lot of times that's a certain sector of it. The other sector, as we were reading, it's for research, personalized medicine, for functions, for those types of things, for academic and research. And then there's also that sector of the field, which is, you know, creating these test tube designer babies. All right, so regulations of polygenic embryo screening. The complex clinical and ethical implications of the polygenic embryo screening emphasize the need for appropriate regulation. In the U.S., embryo testing and selection are unregulated and governed solely by individual clinicians and market forces. This approach contrasts with countries like France, Switzerland, Italy, UK, and Australia, which limit embryo selection to the avoidance of disease characteristics. The UK maintains a list of conditions that can be tested for, while Australia allows for the testing of diseases that severely limit quality of life. A limitation of disease-based model is that the distinction between disease and normality can sometimes be arbitrary, as alluded to in the disability critique above. One regulatory model that has been proposed is the welfarist model, which allows for selection of any trait if it is associated with a reliable increase in expected well-being. This model reflects the evolving nature of embryo screening and to encompass polygenic traits that may not be limited to disease states. However, well-being can be influenced by the intersection of personal traits with social structures. Therefore, a problematic interpretation of this model could allow for the selection of embryos based on sex or race on the grounds that prejudice and discrimination associated with traits can diminish well-being. The welfarist model were to be adopted, it would need to be accompanied by philosophical analysis of what constitutes well-being and a scientific analysis of the relationship between well-being and various genetic traits. This rise in polygenic embryo screening is ushering a new era of human control over reproduction. I would argue DNA production as well, as technological and commercial developments are outpacing ethical and policy discussions. Well, yeah, we can order our own DNA online now, or whatever DNA you want. So, as technological and commercial developments are outpacing ethical and policy discussions, reflecting on consequences of polygenic screening and considering potential regulatory models is critical. So that was fun. I do have another one here to read. Let me just come back over and see how you guys are doing. All right. So that one was fun. That one was from Harvard. That one was copyrighted 2025, but it did reference a date, December 2023. So that one is relatively new as well. This one, let's see. This is from 2017. So this one is older, but we read the newer stuff first. So we'll have to be careful reading this one, but this one is from 2012. It says synthetic DNA synthesis and assembly, putting the synthetic in synthetic biology. Abstract. The chemical synthesis of DNA oligonucleotides We read that word in the first article we read, and their assembly into synthons, genes, circuits, and even entire genomes. And we learned from the other articles that entire genomes are able to be made. Wait, no, was it 99% of the entire genome was able to be made? I think that's what we read. Was it the Harvard one? One of them said 99% of the genome. Okay. By gene synthesis methods has become an enabling technology for modern molecular biology and enables the design, build, test, learn, and repeat cycle underpin innovations in synthetic biology. In this perspective, we briefly review the techniques and technologies that enable synthesis of DNA oligonucleotides and their assembly into larger DNA constructs, with a focus on recent advancements that have sought to reduce synthesis cost and increase sequence fidelity. The development of lower cost methods to produce high quality synthetic DNA will allow for exploration of the larger biological hypotheses by lowering the cost of use and help to close the DNA read-write gap cost. And we know we did that. They've done that already from 2017 to this present day, because we were just reading on that website that you can order your synthetic artificial created DNA from DoorDash. So they got there. So that's good to know about this. And this is kind of fun. Like when you read older articles to you know you're learning a lot when you can pick out what has changed. And so we went backwards in time here. All right, we're moving on. DNA oligonucleotides are typically synthesized using phosphoramidite. I recognize that word from that other article. Chemistry methods, and then assembled into larger constructs by a variety of methods. Recent advances have sought to reduce costs and increase sequence fidelity. And we've done that. They create them on demand, DNA on demand. And it takes about 8 to 12 days to ship. Unless you have something a little bit more customized, it might take longer than that. DNA neither cares nor knows. DNA just is, and we dance to its music. Richard Dawkins. And that was from A River Out of Eden, A Darwinian Life. All right, moving on. It has been said that the 20th century was the century of the atom, in which discoveries on the physical and chemical properties of the elements led to breakthroughs such as atomic energy and weaponry, medical diagnostics, computer, and the microchip, to name just a few. These advances had a dramatic effect on our way of life and helped shape the promise and possibilities of science and technology. In the early part of the 21st century, we are witnessing what could very likely become known as the century of DNA. Oh my God. I think it is the century of the DNA. Yep. As the score to life's intricate symphony, DNA provides the blueprint for biological function. Advances over the last few decades in both reading, sequencing, and writing synthesis, DNA sequences, have made marked changes in our ability to understand and engineer biological systems. These advancements have led to the development of groundbreaking technologies for design, assembly, and manipulation of DNA-encoded genes, materials, circuits, and metabolic pathways, which are allowing for ever greater manipulation of biological systems and even entire organisms. Thanks to next-generation sequencing, technologies capable of generating an estimated 15 petabases. Oh, is that what PB means? I don't know, we're going to keep that word in mind, petabases. So thanks to next generation technologies capable of generating an estimated 15 petabases of sequence data per year worldwide, the current megagenomics era has led to the swelling of biological sequence repositories, with DNA sequences isolated from every organism and environment imaginable. Okay. Associated improvements in bioinformatics. Oh, bioinformatics is so cool. Associated improvements in bioinformatics techniques, and software allow researchers to obtain, analyze, and manipulate these DNA sequences in ways easier than ever. The ever-increasing availability of biological sequence information from all branches of the tree of life has deepened our understanding of biological systems and the interrelated nature of organisms at the genetic level. Next generation sequencing technologies have led to a deeper understanding of human diseases, the microbiome, and the genetic diversity of organisms in our environment. This sequence boom is also allowing for the expansion of scientific disciplines such as metabolic engineering and synthetic biology as researchers seek to use novel sequences in the manipulation of biological systems for the anthropocentric means. That's kind of awesome. All right. In addition, this wealth of information is leading to the development of a variety of diagnostics and therapeutics, which will continue to the long-term improvements of human health. And that, I think, is fascinating, that they are doing this kind of thing in a medicine-based they're doing it for all kinds of reasons, but for me personally, I liked reading about the bodily functions, the processes that different systems do in our body, how they're replicating that, how they're looking at how that works so that they can make personalized medicine. To me, that's just fascinating. This biological sequence data bonanza has been aided by a stream of innovators in instrumentation and techniques for generating sequencing data with high fidelity, increasing throughput and decreased cost, which has contributed to making next-generation sequencing a go-to technology for many applications in biology. The ability to generate large DNA sequence data sets has allowed researchers to create and test large-scoped biological hypotheses using next-generation sequencing technologies that were not possible before their development. Beyond analyzing biological systems at the DNA sequence level, the need to construct DNA from designed sequences has driven the parallel development of methods and instrumentation to produce synthetic DNA at a scale to enable the testing of engineered biological components. DNA reading by next-generation sequencing, when combined with modern DNA synthesis techniques, form the two foundational technologies driving synthetic biology efforts and will eventually instill the predictability and reliability to engineered biological systems that chemical engineering has brought to chemical systems. This being said, our ability to sequence DNA is currently better than our abilities to synthesize DNA in novo. Although new technologies are helping to close the gap. Synthetic biology is emerging as an important discipline with the potential to impact a number of academic and industrial applications, including the creation of novel therapeutics, materials, biosensing, and manufacturing capabilities. Although our current understanding of biological systems is vast, it is still far from complete. adapting exogenous DNA sequences, and the functional components they encode that have naturally evolved within a network of interrelated sequences remains a principal challenge of engineering biology. Currently, the engineering biological systems requires a heavy dose of empirical trial and error to evaluate novel enzymes, expression systems, and pathways for desired function. Typically, this process is accomplished by first designing a desired synthetic biological circuit or pathway using computer-aided design tools. Next, the resulting construct, DNA, is divided into smaller overlapping pieces. Typically 200 to 1500 BP segments. And what was that, the same thing that was up here? No, petabases is not that. Okay, sorry, I had to check. That are easier to synthesize. These DNA components are then synthesized from a set of overlapping single-stranded oligonucleotides in-house or by commercial vendors. The resulting overlapping synthons are then assembled into larger pieces of DNA and cloned into expression vector, and the sequence of the resulting construct is then verified. The sequence verified constructs are then transformed into a cell assay for function. Depending on the results, changes to the construct design can be made and further iterations of the test cycle repeated. This design, build, test, learn, and repeat process has become the backbone of synthetic biology, which in turn has put a premium on automated processes and methods that can shorten the development cycle and increase throughput. Oh, we have a graph. All right, look, this is all the DNA here on the computer. It says sequence manipulation. Then you're going to go to the synthesis. And you have oligosynthons devices. And then we'll go back around here from the computer. You modify, you repeat, functional assay, transform, assemble, sequence, and verify. Somewhere in the middle of this, you do design, test, and build. And look, they have this family tree here. Isn't that crazy? And so you do all this, and then eventually it outputs through the device. Look. Oh, is it going to make it bigger for us? Oh, yeah. That's a little bigger, right? Yeah, that's bigger. Okay. Okay. The synthetic biology test cycle from top to clockwise. Oh, it's going to read it for us. Synthetic DNA constructs are designed and manipulated using computer-aided design software. The designed DNA is divided into synthesizable pieces called synthons. The synthons are then broken up into single overlapping stranded oligonucleotide sequences and chemically synthesized. The oligonucleotides are then assembled together into the designed synthons using gene synthesis techniques. If necessary, multiple synthons can be assembled together into larger DNA assemblies or devices. The assembled DNAs are then typically cloned into an expression vector, and we learned about these vectors earlier, and sequence verified. Once verified, the synthetic constructs are transformed into a cell, and function of the synthetic construct is assayed. Depending on the results, the constructs can then be modified or refined, and the test cycle is repeated until a DNA construct is obtained that produces the desired function. One of the attributes that makes biological systems attractive from an engineering perspective is the fact that biological functions are encoded to a large part in DNA. Therefore, a gross simplification of the biological engineering can be reduced to the design, production, and testing of DNA sequences. As researchers seek to engineer biological systems with novel DNA sequences, the need for custom synthetic DNA sequences has grown. This is particularly true when the sequences to be engineered are derived from metagenomic sequences in which no organism may be available from which to isolate the DNA via other methods. This synthesis of synthetic DNA is often referred to genetically as gene synthesis. And I think the Harvard article we just read, I think they refer to it as gene synthesis. But all of this is in the same arena, that everything we've been talking about here, from artificially synthetic DNA production and genetic babies. They are in the same realm. Of course, there is a wide variety of uses, that genetic babies are just one of the little niches when it comes to this. All right, that was a side note. All right, moving on. Which specifically is the synthesis of gene-length pieces of DNA directly from single-stranded synthetic DNA oligonucleotides. Unfortunately, whereas the cost of sequencing has decreased precipitously over time, the cost of gene synthesis and oligonucleotide synthesis in general has not kept pace. Although technological innovation and market forces are progressively lowering the cost of synthetic DNA, which we know they did. You can order it online. Wasn't it like $2.50 a gene? The cost of gene synthesis is typically directly tied to the cost of the oligonucleotide synthesis from which the genes were made. The cost of oligonucleotide synthesis has not decreased appreciably in more than a decade, generally ranging from 5 cents to 17 cents per base, depending on synthesis scale and the length of the oligonucleotide and the supplier. Traditionally, the cost floor has been carried through the production of gene synthesis products. These synthesis genes currently range in cost from 10 to 30 cents per BP. So it's 100 to 300 for a 1 kb gene. All right, so the company that we were reading earlier is $2.50, right in the middle. So I guess they're market price right now. From traditional commercial suppliers, although companies exploiting newer, lower level cost synthesis methods are starting to bring the cost down. Lowering the cost of gene synthesis would enable the generation of larger data sets by making the cost of gene construction less expensive, meaning that more constructs could be generated for the same cost investment. This would allow researchers the ability to sample a greater amount of the design landscape. With a greater understanding of what does and does not work in a given construct design, a set of general rules could be created that would improve the success of the design process and eventually shorten the design, build, test, learn process. Because the major cost of the gene synthesis is the reagents that are needed for oligonucleotide synthesis. Approaches that reduce reagent consumption improve the robustness and accuracy of the gene assembly process and enable increased throughput have become valuable tools for advancing the usage of synthetic DNA by allowing for lower cost of use. In this perspective, a brief overview of the methods and technologies that have contributed to the production of the synthetic DNA via gene synthesis methods will be highlighted, as well as some of the challenges that have had to be overcome to reduce the cost, increase the throughput, and ensure the fidelity of synthetic DNA. Oligonucleotide synthesis. Modern de novo gene synthesis techniques can trace their beginnings to the mid-1960s. Oh yeah, here we go. We get some history. Oh my God, they're tracing their beginnings to the 1960s. Oh my gosh, here we go. Modern de novo gene synthesis techniques can trace their beginnings to the mid-1960s. And, oh my gosh, hold on, guys. All right. And Gobind Coronara and colleagues' efforts to decipher the genetic code and to reconstitute biological function synthetically. These early efforts focused on chemically synthesizing the ligating together 17 oligonucleotides, which encoded the gene for a 77 nucleotide alanine tRNA using synthetic chemistry still in their infancy, an effort that took more than 5 years to complete. Within 7 years of this work, the first reported synthetic gene encoded the 14 amino acid hormone somatostatin was expressed in recombinant E. coli. Subsequent improvements to the oligonucleotide synthesis chemistries and techniques in the early 80s led to the development of a solid phase phosphoramidite chemistries whose robustness and fidelity allowed for automated methods to be developed to enable scalable oligonucleotide synthesis that is used to this day for the commercial synthesis of the oligonucleotides. Synthetic oligonucleotides for gene synthesis applications are generally synthesized using variations of phosphoramidite chemistry methods, either on traditional column-based synthesizers or microarray-based synthesizers that will be discussed in the following sections. Wow. Oh my gosh. So they started to try to do it in the 60s, and by the 80s, they were able to have some sort of process down to where they could do it successfully, right? But it took a long time, nowhere near as what they're doing today. So the next section is column-based oligonucleotide synthesis. The commonly used phosphoramidite synthesis chemistry consists of a 4-step chain elongation cycle that adds one base pair per cycle onto a growing oligonucleotide chain attached to a solid support matrix. In the first step, DMT protected nucleoside phosphoramidite that is attached to a solid support, usually contained within a synthesis column, is deprotected by the additional triclioacidic acid that activates the supported attached phosphoramidite for chain elongation with the next phosphoramidite monomer. In the second step, the next base is the sequence is added in the form of a DMT-protected phosphoramidite and is coupled with a 5-hydroxyl group of the previous nucleoside phosphoramidite in the sequence, forming the phosphite triester. Third, any unreached 5-hydroxyl groups are capped by acylation to render any unextended sequences inert in subsequent rounds of the chain elongation cycle, and thus reducing deletion errors in the finished oligonucleotide sequences. Deletion errors. That is so cool. They have that with cell. When you're looking at cell functions, it's just a function that some of these things have. I think that's so funny. They named them deletion errors. Anyway, reading on. The fourth step. The phosphite triester linkage between the monomers is converted to phosphite linkage via oxidation with an iodide solution to produce siloethanol protected phosphate backbone. The synthesis cycle then repeats for the next base in the sequence via the removal of the 5-terminal DMT protecting group. After the desired sequence has been synthesized from 3 to 5, the oligonucleotide is chemically cleaved for the solid synthesis support, and protecting groups on bases and the backbone are removed. This process is highly amenable to automation and forms the basis for oligonucleotide synthesizers, which can synthesize 96 to 1536 distinct oligonucleotides simultaneously. These synthesizers typically synthesize oligonucleotides at scales ranging from 10 to 1,000, with a cost ranging between 5 cents and 17 cents per base. The oligonucleotides can generally be synthesized up to about 100 MT with error rates at on or below 0.5%. Column-based coupling yields are generally quite efficient, typically under 99% per coupling. However, the yield of full-length oligonucleotides typically decrease with increasing oligonucleotide lengths of yields for full-length oligonucleotides can be affected by suppressed depurination of the synthesized oligomer, especially at the endozymes during the acid deprotection steps of the synthesized cycle, and becomes especially problematic for longer oligonucleotide sequences. These suppress basic sites can reduce the yields of full-length oligonucleotides by promoting the cleaves of the oligonucleotide phosphate. All right. I don't want to read any more of that. There were a few interesting things in this article that I wanted to get to. Okay. I guess we'll start here. Because DNA is a polymer made up of 4 different nucleotide monomers, gene synthesis and DNA assembly methods are in effect a form of hierarchical polymer synthesis. For synthetic DNA, individual phosphoramide monomers are combined together to create individual oligonucleotides to facilitate the assembly of synthetic double-strand DNA synthon from single-stranded oligonucleotides adjacent are designed to contain overlapping sequences between. And so, side note, I think we were reading that the oligonucleotides are single-strand. So they're able to make those. And so then they're able to assemble them to make the double strand. That's what I'm understanding from here. So they have to be overlapping sequences encoded on the opposing strands of the DNA duplex and are assembled together during the gene synthesis process to make double-stranded synthons from 200 to 2000 BP in length. Sequence-verified synthons can be assembled together by numerous methods to make longer synthetic DNA constructs, encoding entire metabolic pathways or genomes. All right. I did see. Oh, now we're getting into the history of the poly chain reaction. Later in this article, they talk about law enforcement. That's what I want to get to. Shortly after the discovery of the PCR in the 1980s, ligation-free methods for using synthetic single-stranded, I'm just going to say single-strand helix, not the oligonucleotides, that's too hard, were developed to make completely synthetic pieces in DNA without the need for a naturally derived DNA template. Since this time, dozens of different methods have been created to assemble double-stranded DNA from single-stranded oligonucleotides via PCR methods. A summary of many of these methods has been reviewed and will not be reviewed extensively here. All these methods use single-stranded synthetic synthetic strands with complementary overlapping sequences between adjacent single strands to assemble double-strand DNA synthons using thermostable DNA polymerase and PCR. The only differences between the rate of PCR-based DNA assembly methods is in how the subsequent single strands are designed to be assembled together, and the reaction conditions under which they are assembled. The end product of these methods is the same, double-stranded DNA synthon. Of the assembly methods, variations of the polymerase cycle assembly methods is by far the most commonly used to assemble DNA synthons from single strands. Using the PCA, that's polymerase cycle assembly, a double-strand DNA sequence is divided into a single strand, which encoded both strands of the DNA duplex, with overlaps between adjacent single strands that range from 15 to 20 in length. Typically, adjacent single strands are designed with gaps between the forward and the reverse overlapping regions of the assembly. Single strands to reduce the amount of single strand synthesis is required to synthesize a given sequence. Once designed and synthesized, a subsequent single strand for assembly are pulled together in equimolar concentrations and cycled into one pot assembly. Reaction in which adjacent single strands are randomly extended in non-exponential manner by a DNA polymerase to a mixture of single strand extensions, products of various lengths. The mixture is then used as a template to seed a second PCR reaction, in which the desired full-length product is amplified. The assembly mixture in the presence of an excess of the outermost assembly primers. In addition to ligation and PCA-based assembly methods, Gibson and colleagues, as part of their pioneering work on synthetic genomics, have developed both in vitro and in vivo one-pot, single-step methods for simultaneously assembling DNA synthons from single strands and cloning them directly into plasmids, and some variation of these assembly methods are used in most commercial and academic gene synthesis applications reported to date. So that reminds me. So trace transfer DNA are minute damaged DNA that are in fragments. Is this what the next generation sequence technology does? it predicts and basically finishes the DNA sequence? Because if that's true, then they're literally adding to something that's broken, which is never 100% because it's not there, it's missing. You'll never know what the other missing part is. So I'm wondering if what they're talking about here, I don't know for sure, is just generally how this works. It's not just for DNA synthesis, it's just like the general thing that they use for PCR, to amplify damaged strands. So even synthetic strands might not start out or be considered damaged, but you got to put them together and work them up, right? Okay. All right, let's move down here. A lot of this is just science stuff that's probably super boring. Ooh, what do they mean by bar-coded priming sequences? That's fun. Yeah, I guess they have a way to organize and to be able to recall and identify. Error correction. I wanted to get to more history. I think we understand that they're going to do their sciencey stuff to finish the DNA fragments and then assemble them with the single DNA strands to make them a double DNA strand. And they have all different lengths of fragments, which they have to assemble as well. And they are talking about different methods for that. Oh, this is where they summarize it, because it says, one of the hallmarks of synthetic biology is the application of rational design principles to the design and assembly of biological components. However, because it is often difficult to know how well a given DNA construct will work once introduced into a cell, it is often necessary to try several versions of the construct to find which one will work best. Therefore, a greater emphasis on modular design of DNA parts enables the assembly of a greater variety of potential constructs through mix and match, combinatorial assembly of DNA components. Okay. I really thought I saw something in there about law enforcement. All right, we'll read I have another article is where that came from. Sorry, guys. All right, we'll read the conclusion and then move over to that other article that talks about law enforcement. The development of automatable, robust chemistries for chemical DNA synthesis over the last 40 years has contributed to the advancement of our understanding of biology and has laid the groundwork for predictable engineering of biological systems. Synthetic DNA is central to the development of methods to engineer biology, and when combined with massive amounts of sequence data being generated by next-generation sequencing efforts, will contribute to the advancement of synthetic biology towards application heretofore unimaginable. To date, there have been a handful of moonshot demonstrations, such as the complete synthesis of an entire yeast chromosome an entire bacterial genome and the sequence of the subsequent synthesis of minimal bacterial genome, which illustrate the use of synthetic DNA and capabilities of existing gene synthesis methods to accomplish large-scale synthetic biology efforts. These examples, when combined with numerous projects in which synthetic DNA has been used to evaluate functional biological components, create synthetic vaccines, and construct synthetic genetic circuits. And when applied to whole-cell genomic editing, point to a future where the nuances of biological function can in part be understood by design, synthesis, and assay in interchangeable synthetic components akin to synthetic drug development. Personalized medicine. To enable larger scale engineering efforts and to democratize the use of synthetic biology techniques and design principles, recent innovations have sought to lower the cost of synthesizing DNA, single strands, and their subsequent assembly into synthetic constructs ready for use. The use of a microarray and microfluidic single strand synthesis techniques, when combined with automated gene synthesis protocols and improvements in synthetic DNA quality, have made great strides towards this goal. In response to market pressures, commercialization of these techniques and the continued development of other efficient DNA synthesis techniques will further enhance the availability and increasingly inexpensive synthetic DNA. Although the synthesis of relatively small synthons can often be straightforward and accomplished at the undergraduate level, the assembly of larger pieces of synthetic DNA can still pose several technological challenges to even the most experienced DNA jockey. Oh my God, there's DNA jockeys! Problems created by a high degree of secondary structure in the target sequence as well as repetitive DNA sequences can pose challenges to their assembly. Sequences are unknowingly toxic to host cells used for intermediate manipulations of the DNA. For example, cloning for sequence verification or assay of synthetic DNA constructs can also pose problems for the successful assembly and use of some synthetic DNA structures or constructs. Here, too, further improvements in synthetic construct design and in the DNA assembly process will likely enable the successful synthesis of even the most difficult target sequences in the not-too-distant future. What started with synthetic tRNA gene more than 40 years ago has led to the recent synthesis of the first minimal bacterial genome and a deep appreciation of complexities and challenges of engineering biology, much of which still remains to be explored in this century of DNA. The question remains, what's next? Oh my God, it said here in the last paragraph, creating DNA with the relatively small synthons is as straightforward that an undergraduate can do it. That is crazy. And so they named this the century of DNA. I've always been interested in DNA, and I've been reading about it for decades now, and I've learned a lot along the way, and it surprises me how for a lot of the public and some juries, they've just decided DNA is DNA and it just doesn't evolve. Well, science has never stopped, has it? Do we know anything in science that just kind of stops? The only things I can think in science that just kind of stopped is when we learned new techniques that are better or faster or just more practical, and then we go in that direction. So yeah, DNA science is never going to stop. It's forever changing. And maybe DNA science wasn't talked about for a long time because of the ethical implications of designer babies and designing DNA. It is controversial, so maybe that's why DNA stopped being so mainstream, and that maybe there was a gap between what the public was learning and what was actually happening in science. That could be. I think things are changing now. Where, as we saw in the Harvard article, that they had a study in there that said 70% of the public was okay for synthetic DNA or DNA sequencing for health conditions. So for the health aspect, that's the whole reason why I research it and enjoy it. I think the public is growing towards being ready for that. Any other functions of this could still be controversial. I mean, I don't know. But it's always going to be used for research and study. I mean, that's why we have a field of research. And I'm constantly reading all of the research they have on this kind of stuff. So they will use it for that. And we're going to get more pharmaceuticals, and they're going to study more of this. I mean, with every industry, there's always a sector that calls for concern and discussion. And maybe that's why DNA kind of fell out of the public view, is because of all these ethical concerns and where DNA technology is leading. I think that could be why. All right. Is this okay? So this one is from 2022. All right, so this one isn't that old. Is it 2 years now? Too big. And this one is called. This is from a book. Oh, it's from dentistry. Why are we reading a dentistry book? Well, it says regulation of biological agents and biotechnology. I think the book is called Synthetic DNA, Synthetic DNA, and this section is called Synthetic Biology. Yes, this is the one I think about law enforcement, because I see Crime and Research Institute. Where did I? All right, I'm going to say view this chapter. It's got to be the first one. Oh, it gave me a new page. Okay, I don't think that's it. It's definitely this thing, but then when I click on it, I don't think it goes to the right thing. Yeah. I need to share more. Yay! Yeah, this is it. Yep. Okay, where do we want to start? Do we want to start at the very beginning? It's really long. All right, so this is it. Synthetic biology, also referred to as SynBio, is a combination of engineering principles and computer science with molecular biology, enhancing scientists' ability to manipulate microorganisms or the genome. Synthetic biology can be used to create synthetic genomes from viruses and bacteria, as well as genes to produce toxin proteins. To produce new materials. Synthetic biology also allows for enhanced manipulation of genomes for the production of novel biological components or organisms. One example would be rapid production of medical countermeasures or new materials. Synthetic biology has been used in medical science to produce novel countermeasures or treatment for disease. For example, the United States Food and Drug Administration approved human therapeutic made using gene editing was insulin. To produce synthetic insulin, recombinant human insulin is produced in either E. coli bacteria or yeast. The therapeutic advantage of recombinant human insulin is the changes in the amino acid sequence. Okay. Since 1983, when synthetic insulin was brought into the market, biotechnology has been used to produce a number of recombinant therapeutics. Synthetic biology has also been used in preparing novel vaccines. The World Health Organization declared COVID-19 a pandemic on March 11, 2020. And there was a global effort to produce safe and effective vaccines. Okay, we know. In 2002, synthetic poliovirus was constructed. In 2006, a group at the University of Alberta in Canada synthesized the horsepox. Oh my gosh, they're making diseases behind the closed door. For synthetic genetic material ordered from a commercial company. Oh man, they ordered the DNA. They ordered horsepox DNA from that from that article we read earlier. You can get it in 8 business days. These researchers ordered overlapping synthetic DNA fragments, about 30,000 base pairs, which they litigated together to form 200 to 12,000 base pair horsepox viral genome. They made a whole genome out of that virus. The genome was transfected into a cell that was also affected by a different pox virus, which integrated the synthetic genome and produced infectious horsepox virus. This research raised security concerns because horsepox is no longer found in nature and is closely related to smallpox virus, which was declared eradicated by the HWO in 1980. The production of synthetic horsepox viruses has raised the issue that this technology could also be potentially generate smallpox virus. It was assessed that it would take a small scientific team about a half a year and cost $100,000 to generate synthetic smallpox virus. This initiated further discussion on the terrorist and rogue states using biotechnology to produce synthetic biological weapons of security concern. However, ordering a smallpox genome fragments, oh my God, can you imagine ordering smallpox or horsepox from that company? And like breaking during the mail? No, I don't think so. It wouldn't break. I think they take into account transportation, and it's probably environment control, temperature controlled, but like I could just see it happening. However, ordering smallpox, you can order smallpox in the mail. It's fine. It's totally fine. Genomic fragments from commercial suppliers should raise tripwires with companies that would be reported to law enforcement agencies. Here we go. The generation of synthetic smallpox genome and infectious virus is internationally regulated, and countries have also introduced regulations regarding access to biological agents of security concern. Synthetic biology has been used to produce new materials. For example, the Defense Advanced Research Projects Agency, DARPA, has several projects using genetically engineered microbes for the production of materials. For example, the biomanufacturing, survival, utility, and reliability beyond Earth, called Be Sure program, aims to explore the potential to use biomanufacturing for novel in situ manufacturing in far forward locations such as space. The National Aeronautics and Space Administration, NASA, already uses additive manufacturing, 3D printing in space. They use 3D printing to print their diseases? What? But the Be Sure project would take this one step forward to elevate supply chain issues in space operations by collecting data on the microbial utilization of the space-based alternative feedstock. All right. So they are using synthetic DNA in space as like a farm of sorts. I think they even have a farm. What was it? Is it in like Switzerland or something, or Sweden, where in case like the world ends, there's everything, all the life here is preserved there, so that if anything happens, it could be reestablished. I don't know what the purposes are that the space would use it, but it's I think to be expected that they would be doing science up there, sciencey stuff, and maybe it's more practical to do everything synthetic instead of taking real biological things up there. Okay, so that's cool. They probably have a farm and they're testing and doing all kinds of the same stuff we're doing here. They're doing up there. It's awesome. And so they're finding supply chain issues with that, of course. There are space-based alternative feedstocks, optimization of microbial growth in variable gravities. Oh yeah, science experiments. And mitigation strategies for identified effects of galactic cosmic radiation on microbial growth and bioproduction. Awesome. The advancements in synthetic biology have enormous benefits to medical science, military, and other industries. However, the dual use potential for synthetic biology to be used for malicious purposes requires monitoring and regulation for its use. The following discusses recent advancements in biotechnology that have potential dual-use applications. Genome engineering refers to the process of making targeted modifications to the genome. This includes modification to regulation of non-coding regions. It's my understanding that law enforcement do use non-coding regions, and it's easier to find a match with those. I don't know what the specifics are, how that's done. There was an article we were reading one of the few lives ago that were saying that this non-coding DNA is used by law enforcement, and it is easier to find identification with it. So back to the article, this includes modification to regulation of non-coding regions, for example, epigenetic markers, promoters, or the outputs of the transcription, for example, RNA, which may result in changes to the translated protein. The technology for genome editing has advanced substantially in the microorganisms through molecular biology techniques to transgenic insertion of genes or knockout, the deletion of genes, mice to study gene function, to target mutations of the mouse genome using N-ethanol and nitrosourea, ENU, the study of the function of novel genes. The type 2 clustered regulatory interspace short palindromic repeats. Oh, it's CRISPR. Associated proteins Cas system is encoded by bacteria and acts as a defense mechanism against bacterial phage, which is viruses that infect bacteria. It also facilitates plasmids, small circular bacteria DNA that often encodes genes for antibiotic resistance or toxins important to bacterial functions. Since the discovery of CRISPR-Cas complex, it has been used as a powerful RNA-guided DNA targeting platform for genome editing, transcription, epigenetic modulation, and genome imaging. This advancement in technology has been significant for biotechnology as it allows for precise manipulation of virtually any genome sequence. We were reading that today. In cells, organisms, or whole genome that is specified by a short stretch of guide RNA. This resulted in determining gene function in diseases, correction of mutations that cause genetic disorders, such as many forms of cancer and neurodegeneration, as well as sickle cell anemia, cystic fibrosis, and Duchenne muscular dystrophy, viral infections, immunological disorders, and cardiovascular disease, and activations of oncogenes, genes that enhance cancerous cell production, or activation of cancer suppressor genes that have been deactivated. That's what they were doing in this cancer study I was reading. They were learning how to activate those suppressor genes by studying how those suppressor genes interact with the genes or the cells that enhance cancer cell production. You know, just think about, like, I don't know, these little things going to battle. And you can watch them, like, you know, interact with each other, and how one can get in the other, and how they affect each other, and then, you know, create a pharmaceutical from it. It's so amazing. I was learning all of these different terms. It's medical terms for, of course, you know, terms that are more than what you learn in physiology and anatomy. And it's specialized stuff that, like, you know, and then they're able to create that they can watch your DNA, and they can watch your battle going on with your cancer genes, and then they can individually create a medicine that works with what is going on in your body specifically, called personalized medicine. I don't know if we're there yet, but that's in research now. And it's awesome. So, like, I think that was, like, the newest thing that I was reading. But I'm sure since I was reading that, there's so much more. There's so much new things that are coming out that's probably old news by now. So where did I leave off? Or activation of cancer suppressor genes that have been deactivated. Like, they're learning how to activate them too, and identify drug targeting disease-resistant genes. It is the properties of CRISPR-Cas as high-efficiency cell-specific nuclease, together with the possibility of highly multiplexed modifications, have opened up a broad range of biological applications across basic research, biotechnology, and medicine. The high-efficiency site-specific targeting is due to the Cas natural function to target phage and plasmid sequences, which can easily be replaced with a target sequence to allow Cas to nuclease the specific target regions of a gene. This allows CRISPR-Cas to be used as a RNA programmable DNA targeting and editing platform for which specific and multiplex genetic engineering. So we're able to code DNA now as well. They wanted to learn the specific way that these cancer deactivation genes are coded so that they can do the counter code. So that also, not only are we able to synthesize the DNA, we're able to code it as well. So it's beyond just creating synthetic DNA. It's manipulating it too for medicine, which, I mean, that is amazing. Absolutely amazing. That, like, I don't know, like, I would never have thought that that would ever be possible, and like to just be able to read it is so exciting. Okay, wait. Yeah, here we go. Here. Programmable DNA targeting and editing platform, more specific multiplex genetic engineering. Multiplex targeting by Cas is achieved by introducing a battery of short guide RNAs rather than a library of large, bulky proteins. Oh, they've learned strategic ways to do it. Another advantage of type 2 CRISPR-Cas is the existence of many natural variants that can be used to efficiently target, edit, modify, regulate, and mark genomic loci of a wide array of cells and organisms. Awesome. Obstacles for CRISPR-Cas use in medical treatment include the potential unintentional mutations due to binding to sites with sequence homology to the specific target sites. This is especially important as the genome editing leads to permanent modifications within the genome, enhancing the effectivity of the specificity of the CRISPR-Cas sequence targeting is essential for widespread use for genome editing for human therapeutic use. Yes, they're editing DNA, they're editing how the DNA is coded, and they're all doing it for therapeutic use. But as this article says, there's always, there's multiple applications. Someone could be using it for good and someone could be taking advantage of it. It just depends, and that's the nature of these types of things. The ability for CRISPR-Cas to specifically target genomes, human, animal, plants, bacteria, and viruses, and multiplex to target a variety of genes, there is potential for malicious use. Therefore, CRISPR-Cas can be considered a dual-use technology, addressed in technical and ethical issues related to the safe, responsible use of CRISPR-Cas, including the potential to edit the germline cells in embryos where the genetic changes would be passed on to future generations versus gene editing of the somatic cells where the trait is not inherited. Although there is a concern regarding genetic engineering for treatment of disease or disorder versus human augmentation. Okay, you see how personalized medicine, how they're studying different functions of different genes and to help with therapeutical things, because that's so tightly interwoven with these controversial issues, especially with genetically engineered babies. As I'm learning about the medical side of all this, and since it is very close with the genetic engineered babies. I'm also learning a lot about that too. But that's one of what they're talking about here, that there are multiple uses for these things, and since they're so interrelated, it just by happenstance, I've been learning a lot about that too, because I think they're similar things that are just on a different sequence. Like they're so related to each other that they're always written about together, because the same processes that they use, that are, say, in a scientific way, the way that we look and we're able to examine and best standards for examination and those types of things. I think across the board, scientists kind of follow the same scientific method or similar. And so you'll see a lot of it written from that perspective. But since it's dual use, a lot of these things are in the same articles, as we've seen, that they are very closely related. So by accident, I've been learning a lot about genetic and engineered babies just because I'm learning about medicine. They just happen to be together. So here we go. The ethical and clinical issues have been raised in 2018 when the first genetically engineered babies were created in China. Did anybody hear about that? I don't remember hearing about that. So if we have a whole genetic baby that was created in China, like for real on the books, it's not just these like mystical, like weird labs where you can like order stuff. Yeah, DNA can be created and manipulated. And that is just day one stuff. That's not like creating a whole body that's made up of DNA and cells. Wow. Let me know in the comments or in the chat if you heard China made the first genetic engineered babies. Well, they might not be the actual first ones, because we know these clandestine, like non-government NGOs, they probably have done it, but this is the first one that's like documented for public view. Wow. Okay, we're gonna have to have another live about those real genetic babies. Okay. Oh my God. Okay, so CRISPR-Cas technology was used to edit embryos of twin girls to disable the CCR5 gene, which is involved in the molecular pathway for infection of viruses such as HIV and smallpox. At the time of these experiments, there was no regulation of CRISPR-Cas. Concerns have been raised regarding genetically enhancing of children to be protected from infectious diseases such as HIV and smallpox. It is an ethical gray zone between treatment and enhancement. I would say they are correct with it. It's an ethical gray zone because it's so closely related to like creating people that like you're essentially using the same functions to study the person to give them medicine, then it's the same things to actually create a person. So I guess they consider the medicine a gray zone, and like that other stuff is in the red zone. Like I don't know, yeah, I know, you know what I mean. All right. Okay, so questions have been raised regarding if this was human augmentation, as it was not attempted to cure disease or disorder, but provide a health advantage to engineered embryos when developed. I don't know if they've gotten to that point yet where I think they can get close, like because they're modifying things. They use that Punnett square, and I think they've been able to get close. I think it's not a complete total exact science. Like you can't just like print out exactly everything that you want. They might be able to do that. I don't know, but I think there's more a little bit. If there's still nature in there that that we can't access, but we can influence it in a way that we're able to modify and create and manipulate. So when it says here that they can influence. I think that's the extent of it. They are influencing to the point where they're gotten really close to being able to just do more than just influence it. But I don't know for sure. That's just what I'm getting from this. All right. The editing of the human genome in embryos is illegal in several countries around the world. In the U.S., it is regulated by the FDA. So regulated. Does that mean there's laws? It's lawful to do certain gene editing, certain DNA synthesizing? Oh, geez. So the guy who's like responding to Sherlock Holmes, he doesn't even know it's regulated here in the U.S. Okay. So who is the Janko? Is that who created the babies? Yeah, Janko. Janko. Well, no, I'm not saying it right. Anyway, the guy who created babies was placed under house arrest in December 2019 and convicted in the criminal justice system for deliberate violation of medical research and regulations and rashly applying gene editing technology to human-assisted reproductive medicine. They got him. So, like, who's doing, like, human experiments? Like, I guess that's kind of what you're doing when you're creating DNA from scratch. I mean, I don't know how it works, but they put him on house arrest. That's so dramatic. Several other researchers involved in the experiment were imprisoned. Oh my God, I didn't hear about this at all. In April of 2022, he was released from prison. Well, he was on house arrest, right? And I guess then he went to prison. In the U.S., the first adult has been treated. Okay, so treating is different than creating a baby. Didn't China create the whole babies? Oh, they were genetically engineered babies. Okay, and so in the U.S., the first adult was treated using the CRISPR-Cas to genetically modify somatic cells and cure sickle cell anemia. The clinical trial has now been extended to children under 12 years of age. So, yeah, they're manipulating the DNA to be able to do that. So they have to be able to create DNA to look for. So when you are creating DNA synthetically, you could either do it with a DNA template, which is what they would do in the medical side, because they would use the subject or the patient's actual DNA, so they have their DNA template. There are other uses for it where they can create it from scratch. They don't need the template anymore. And so, those two things are both considered DNA, synthetic DNA, to artificially create it. So the clinical trial approved by the FDA, however, genome editing of the germline cells that result in inheritance of edited genome have not yet been approved. So they know that when you have genetically engineered genes, whether that's from the template or you're actually like creating people and you're creating DNA from not a template, they know that it can cause changes in inheritance, which tells me that there is more going on behind the scenes. And, from a science standpoint, why not? As long as it's ethical. That's creepy, though. It's real creepy. Okay, where was I? Right, so the result inheritance of edited genome have not yet been approved. See, it's not yet been approved, but they did it. Therefore, although CRISPR-Cas has now been approved for use in the somatic cells in the U.S., there still is a need to regulate the use of the technology, and there is the potential law enforcement investigation for this technology to be misused. Here we go. I want to hear about that. That's the whole reason why I wanted to read this article. I briefly looked at it. I was like, yeah, I definitely want to read that because I thought there was history of it and then stuff about law enforcement, but we got a whole lot more out of it than I thought. All right, where were we? All right, several authors have raised the issue of nefarious use of CRISPR-Cas and its dual use potential due to the ability to modify microorganisms, plants, animals, and humans, and or produce engineered bioagents. Which I think are people or animals. Animals. There was that cow, the first one, that was for public consumption. Other authors have assessed that the risk of misuse by inexperienced terrorists is limited due to CRISPR-Cas9 having several limitations, such as sequence targeting, and would present a number of technical challenges to be misused. Less technically challenging is do-it-yourself biology, which is a growing biotechnology movement where individuals, communities, and small unregulated organizations undertake genetic engineering, biohacking, gene amplification, and detection technologies in formal improvised laboratories. So is biohacking modifying a DNA template? Because biohacking seems to me that something already exists and you're manipulating it. So I do think that DNA sequencing or synthetic DNA, it does cover a DNA template, and it does also cover things that are just from scratch. So biohacking, I don't know, I guess it could be considered either. Okay. Oh God, where did I go? Okay, there has been concern that do-it-yourself biologists now have access to CRISPR-Cas technology, with several commercial companies supplying online. Yep, we know, you can order it. $250 a gene. Such as do-it-yourself bacterial gene engineering CRISPR kit for around $200. In the US, the FDA has highlighted that do-it-yourself genetic engineering is illegal and risky, and is a requirement. And there is a requirement to monitor the ethical and security concerns of this technology being used. Here it is. There it is. This is the moment I've been waiting for. Hi, Preston B. Preston B says, I've never heard about a genetically engineered human, only animals. Yeah, Preston B, they've been doing it for a very, very long time. And they've been doing it with seemingly endless cash flows that are non-governmental organizations. And we would, as the public, we would never hear about them. And they work closely with government too. So it just depends on what experiment or what their target project is. Okay, so here's the moment we've all been waiting for. Concern has been raised that criminals will be able to modify their DNA to avoid matching DNA profiles stored in a national and international database. I also think you could flip that around. Say law enforcement biologically engineer their sample to match their suspect better. DNA profiles used in forensic science and for upload onto DNA databases such as the FBI Bureau of Investigation Combined DNA Index System, CODIS, is based on short tandem repeats that are repeat non-coding sections. Non-coding sections. See, they do use non-coding sections for identification, and I thought non-coding sections have more health data in them. I'll have to read that again because I thought we read an article where law enforcement does like to use non-coding sections, but non-coding sections are also where diseases are. So if that's the case, yes, law enforcement is 100, if it's the case, they're 100% looking at our health data when they identify someone with DNA. Okay, so which repeat for 100 or 400 base pairs. The CODIS core loci amplifies 20 STRs plus amylogen and polymerase chain reaction PCR to generate a DNA profile. For a criminal to modify their DNA using CRISPR-Cas technology would require modification of the STR to either eliminate the STR or change the size of the STR. That has been referred to as generation of ghost DNA profiles by modification or alteration of the target genetic markers. I don't think the average criminal is thinking about changing their DNA. That's just a no. I don't know if these I'm sure these professional criminals have been able to do it for a while, but no, not your average person. I mean, where do they get? I mean, I guess maybe they just admitted you can buy all this stuff online to change your DNA, change your markers. That is terrifying. What did I say? $200 you can change your STR, your markers. The issues with modifying STR markers are that they are repeat sections of various lengths. Targeting the STR with CRISPR-Cas would be difficult, and there could be off-site targeting. Alternatively, targeting the flanking region of the STR could be used to delete the STR. It is unknown what effect this will have on the function of the downstream gene, that the non-coding region that encompasses the STR is involved in regulating. The other issue is what cells would a criminal need to target? They don't know. Okay, if there's a criminal that can learn that or is doing that, you've got something else entirely. You've got some professional assassin. You don't have, there's no way. I don't believe it. I don't believe that there's these criminals that aren't professionals that work for some organizations. There's no way. Would they only have to repeat skin cells? Well, wait, would they only have to target skin cells, semen, hair, blood, so that the DNA profile left at a crime scene is altered, or could they target the buccal cells to that any reference sample collected would not match a crime scene sample? Okay, not even Brian Koberger can do that. They think Brian Koberger is some mastermind. He can't do that. No. Okay, let's think about it. If the general public and the juries don't understand DNA whatsoever, how are criminals going to do it? They're from the same population. I don't know. This is a stretch here. I'm thinking this is the other way around, that law enforcement is doing this, to beef up their case. No, this is not happening. The validation and verification of massively parallel sequencing utilizes SNPs, means that this technology can be used if ghost DNA profiles were suspected. I wonder how they're using the term ghost DNA, because I always thought it was from ancestors that we don't have a fossil library from. Like if you look at the Neanderthal and the Denisovans, and I think they were saying North Africans, you can look at their genomes, and you can say that they inherited genes from an unknown population. That is what they call ghost DNA. But in this paper, I don't think they're referring to that. How are they referring to ghost DNA? SNPs means that this technology could be used if ghost DNA profiles were suspected. Oh, it's ghost DNA profiles that the do-it-yourself, you buy your own DNA profile from online? That could be too. These ghost DNA profiles, who knew? Huh. Okay, another advantage of massively parallel sequencing is that it would be more difficult for criminals to modify SNPs, since significantly more SNPs are targeting the massively parallel sequencing than STR. And thus, to target SNPs will require multiplex targeting with CRISPR technology. Now, I'm wondering if when Ian Taylor said that between Ortham and the FBI, the SNP profile doubled in size. I wonder if the police did this. I know, that's crazy. Why would the police ever do that? But I certainly don't think criminals are smart enough to do this. I don't even know. I'm so confused by this article here. Okay, so CRISPR-Cas biotechnology also has the potential to be utilized in forensic science. For example, it has the potential to be used for enrichment of targeted STR markers in low copy contributors. That's low copy trace DNA in degraded samples, which also is transfer DNA. It's all degraded, all trace is degraded, and mixed samples. This overcomes the issue that occurs in some DNA profiles. When stutters, we learned about stutters, it's any fabric or fiber that masks itself as an allele. And it could make things look like they match or they don't match, and it's not an accurate representation. One or less of the STR motif is amplified, changing the DNA read and resulting in artifices. In these cases, it has been demonstrated that CRISPR-Cas technology can be used to make copies of specific regions of the genome by cutting out the STR through targeting of the flanking regions. Unique molecular tags are added to the excised STRs, and they are then sequenced without the need for PCR amplification. This technique also allows for the identification of minor contributors in a mixed profile. Demonstrated that by including SNP, about 100 bases from the STR is excised using CRISPR-Cas. Allows for the identification of the minor contributor by massive parallel sequencing. This novel combination of CRISPR-Cas, STR, and massive parallel sequencing is referred to as the STR-seq. That's interesting that we are reading a synthetic DNA article, and they're bringing in law enforcement. And they're talking about dual use. So police are using this for multiple applications. Gene expression and function is dependent on the context of transcription. Genes can play different roles in cell-specific functions or pathways. CRISPR-Cas have now been used to generate pools of cells with diverse genetic perturbations, which can be subjected to selection followed by sequencing to assign phenotypes and genetic permutations. Gene permutations allow for the exploration of the gene function in specific cell context. Perturbation sequencing, perturb-seq, has been used as a novel method to map the transcriptional effect of genetic perturbations. It combines CRISPR-based genetic screening with information-rich single-cell RNA sequencing phenotypes. One of the advantages of PerTube-seq is that it can be applied at the genome scale for the systematic prediction of the gene function and the study of the complex cellular phenotypes. This enables massive parallel function genome mapping, which has now been performed at the genome scale single-cell targeting gene in CRISPR. Interference CRISPR-I across 2.5 human cells. Transcriptional phenotypes have been used to predict the function of poorly categorized genes and covering new regulators of ribosomes, biogenesis, transcription, and mitochondrial respiration. The PerTube-seq not only allows for the assignment of gene functions, cell transcriptional phenotypes allow for in-depth dissection of complex cellular phenomena from RNA processing to differential. This provides a rich phenotype maps to provide detailed information on gene and cellular functions. All right, last little bit. In the future, the use of massive parallel functional genomic mapping can have a potential in forensic science applications for phenotypic analysis of transcription from cellular material left at the crime scene. For example, determining complex cellular phenotypes to go beyond determine someone's externally visible characteristics, but to also predict emotional state perpetrator victims. That is terrible. Because please don't tell me that's coming down on law enforcement. There's a lot of health things that go involved in emotional expression, even in hyper states. And even if they are looking at our phenotypes, certain phenotypes are susceptible to certain diseases. I still think law enforcement is looking at our health data all day long. Even the data that I guess people have categorized as actually not being health data, I think that actually is health data. But I don't know. I think they're going to continue. The more that law enforcement is able to look at, the more likelihood they're looking at actual health data. There's no way that this isn't health data right here. Oh my God, they need regulations of that ASAP. The circadian rhythm to determine time the biological material is left the same. The circadian rhythm is health data. So it's just out right there that it's fine for police to look at health data. That is terrible. The opportunities for PerTube-seq also comes with potential misuse, as an understanding of genotype phenotype provides information on important gene functions. Coupled with CRISPR-Cas9, it could be used to inhibit gene function in organisms or for insert a specific genes into an organism to alter the genotype and change phenotype. The history of malicious use of biological agents taken together with the enhanced potential for genetic engineering of bioagents highlights the need to regulate access to bioagents and use of biotechnology. The next section provides an overview of the current international and national regulatory framework. Oh, that kind of like, and come back and say hi to everybody. Yeah, I don't know. That's surprising me that they're talking about law enforcement in a genetic DNA engineering article. That's a little scary. All right. Wow. Well, that was fun. So this was to back up Sherlock Holmes. He's 100% correct when he's talking about that DNA can be created from either a DNA template or just from nothing. They can just create it. They don't need a template whatsoever. And I don't know if you guys have seen his reaction video. I will link both of those videos in the description. All the articles that we read here today are already in the description if you're interested in reading more about those. Synthetic DNA has been the thing. They've already created people and it's already been public. So whoever that guy is, that's just like, no, you can't do it. I read scopes. No, he's seriously misinformed. So yeah, they can create DNA. So this was fun. And by no way am I saying that they created or genetically modified Brian Koberger's DNA. I'm just saying that when I'm reading this about DNA, designer DNA or synthetic DNA, I'm just relating it to the case and giving possible examples for discussion purposes. It's not because I think they manipulated DNA like that. So, I mean, I have no idea if they did or not. I'm not going to get that much into that because that's just too much for me. But I do know that there's something wrong with the DNA, and their processes and their standard operating procedures might be off, which means the interpretation of the DNA would be off as well, because that's part of it. It's more than just looking at the genetic material or the output from the software that they use. It's also, how do you run your lab? What are your protocols? Because if any of that is not functional, then yeah, your entire system of how you interpret DNA for that lab can produce errors in results. And that's dangerous if your lab is skewed from that kind of thing, and then there's a period of time where all of the results that you're providing to law enforcement are incorrect. That's bad. Or if you're doing a scientific study and you're using a lab, a DNA lab for one of these studies or whatever you're researching, they want their results to be accurate to be able to publish. So they need to make sure the labs that they are using and the standard operating procedures that they have are accurate to keep the results as accurate as possible, to be able to have the empirical evidence to show these are the results, these are our theories, and this is how we can rely on it. So, yes, I don't know if people are going to leave this live and go, oh, she thinks that his DNA was genetically engineered. No, these are just discussion purposes. I don't think that at all. I have no idea. I'm more of a systems approach type of person where I want to know what the systems are. Because the systems, if you're working within the system, as we've seen, that can go wrong too. And like they have to, like the silo effect for that one lab in Queens, Australia, the way that they are operating worked was not efficient. They were not getting accurate results. But they have that empirical data because they did have current operating procedures, which they were able to use to be able to change them. If they didn't have any of that documented, they couldn't have gotten to the point where they're saying, all right, this type of model that you're using in your lab is not working, it's not yielding accurate results. And so we have that. So even if an institutionalized standard, that they can still recognize if that's not working. And it's by nobody's fault. That's just, they were following the rules until the rules changed. But then there's also times where maybe accidents happen and, you have to be able to account for these things that might throw you off course to keep your lab running efficiently. And that is just as part of the DNA interpretation, the actual genetic material or the outputs from the software. And so those types of systems that keep the lab running, that's where my interest has come in. What is the process that could happen or did happen? Not that it actually did. processes are completely different than the actual science or what actually happened. You're just looking at empirical data. You're just looking at data, basically, just what's documented, what happened here, or what could have happened here. That's it. So, yeah, designer DNA, designer babies. They're so closely related that I've learned a lot about designer babies because I'm trying to learn about the pharmaceuticals, the personal medicine, and how it relates to synthetic DNA or genetically engineered DNA to help with diseases. For whatever reason, they use similar systems, and they're looking at similar aspects of the same thing. So that was just by accident that literature is just there with the literature that I want to read. So, yeah. I guess I want to play one more thing. It'll be perfect for this. Okay. Yeah, that's good. Okay, I want to share this.
Who says DNA can sparkle bright? Strands of stardust, pure delight, golden genes and cosmic.
I don't know if you can hear that because I tried to play one of my songs before and it didn't work. Oh, you know what? I may have sent it to myself. Hold on. Did you guys hear that song I played for a second? Oh, I don't think that worked. Oh, you can. Oh, good. Okay, I'm going to start it again. Thank you, Preston B. All right, awesome. I'm going to play this again. This is is it my newest one? I have two new ones. I don't know which one is newer, but they're both new.
Who says DNA can sparkle bright? Strands of stardust, pure delight, golden genes and cosmic beams shining in our bloodstream. We're made of fire, made of light, born to shimmer in the night. Dripping diamonds laced in gold, a story in our helix tone. Glitter, glam, and DNA diamonds, neon veins and hearts ignited. We don't break, we crystallize, shining bright in every light. Chromosomes like chandeliers, dripping magic through the years. Molecules in haute couture, flawless fears forever pure. We're made of fire, made of light, born to shimmer in the night. Dripping diamonds laced in gold, a story in our helix told. Glitter, glam, and DNA diamonds, neon veins and hearts ignited. We don't break, we crystallize, shining bright in every light. Glam's not just a state of mind, it's coded deep inside our kind. Falling stardust in our veins, we're couture in every frame. So who says DNA can't shine? We're designed by design. Glitter, glam, and DNA.
I love that one. That is so good.
Process.
Awesome. I think that's all I have for you guys. I think that was fun. Oh, before you go, let's just look at this again. Oh, where you can order it. Where did it go? Here you go, ordering. Ordering. You can order jeans in tubes, jeans in plates. And it's, oh, well, this one is $25 a jean. This one's $120 a jean. This is all synthetic DNA that they made without a DNA template. Now here you go. Advantages to onboarding your vector with IDT. So you submit your vector, that's your DNA sequence that you made. Put it on your credit card. They put it in a box with these are the test tube ones. They have quality control and manufacturability. They send it to you, and then they will email or mail you a notification that it was set. Oh, look, up here, it has, oh, look, see, they do cloning. They clone it. Oh, look, see, this one for additional options for genes, that takes 3 extra business days, 4 extra business days. Up here, let's say you're going to order a test tube. If you get a mini gene, it's $125. It takes 8 business days. But if you want like this big gene, oh, look, it's 30 cents. Oh, it takes 12 business days. They also have on here, you get 40% off. I guess your options for genes, which is down here. Additional options for genes. Okay, you get 40% off that. You add this promo code, gene add-ons. And then you can also get $500 off when you onboard your own vector. And this applies to your first gene synthesis order after onboarding. Promo credit will be issued upon completion of the IDT vector onboarding, but that expires. Oh, they both expire at the end of the month. So get your DNA while there's this 40% off. Will DDT store it? Yeah, because you have to create your own online account and they'll keep it. Why don't your cloning vectors contain multiple clone sites? By not having a set of multiple cloning sites in our vectors, customers are able to choose the optimal restriction site required to subclone into their choice destination vector. You should add a restriction site to your sequences when placing your order. Oh yeah, what are the optimal storage conditions? So while dry, they should be stable for up to 2 years. And you should store the DNA at negative 20 degrees Celsius. And if necessary, avoid more than 2 or 3 frozen thaw cycles. Okay. Why does the sequence data for my custom gene have errors? Even with clonally pure samples, cloned samples, next generation traces rarely show 100% sequence identity. This is because errors do occur during sequencing reactions and the subsequent data analysis. The error rates are typically between 0.1 and 0.01, but they can be locally elevated in regions of extreme GC content and secondary structure. Finally, some small subpopulations of the sequence variants can arise during propagation of E. coli. So, this is cool. Are custom genes delivered as linear fragment or in a vector? IDT genes and many gene sequences are delivered as a double-stranded DNA, cloned into an IDT vector. Okay. Awesome. Oh, they verify that IDT gene customers are legitimate scientists that engage in bio research, and they gotta verify you're a real scientist before they deliver. Well, here we go. In October 2010, the United States government issued final screening framework guidance for providers of synthetic double-stranded DNA, describing how commercial providers of synthetic genes should perform gene sequence and customer screening. IDT and other IGSC member companies support the adoption of the screening framework guidance, and IDT follows that guidance in its application of the harmonized screening protocol. Interesting. Examination, transporter of fluorescence polarization, precise template independent correction, in vivo selection, all right. All right. Thank you, everybody, for being here. This was a long one. Have a good night, everybody. Bye.